Hello and welcome. And first, thank you so much, Tetsu Paul, for the lurk. Much appreciated. Hope you have a great time at your sister's and hope you're having a great weekend. And also hope that you're having equally as good, if not better, weather than I'm currently experiencing. Right now, the weather is great here. I'm in Montreal. Pretty much summer is here. Took a while to get here. Definitely some ups and downs, and like a random ice storm, and a quick turnaround, and then some rainy, some couple weeks of rain, and then now here we are. But now that I said that, I'm sure whatever the weather is just gonna get like next week. But regardless, thank you to anybody tuning in. So I was supposed to stream yesterday, as mentioned in the Discord, but not really my content. It's supposed to be on Wick's panel, a discussion panel, trying to turn Wick to a leftist as opposed to a centrist. But that got rescheduled to Tuesday. So far it seems like it's confirmed, which is great. And hopefully I will be streaming it on my end. Probably going to stream it to YouTube as opposed to Twitch, again, to separate content. And also, as mentioned in the Discord, I've reset my modem to factory settings. It's no surprise. If you've tuned into the streams, you've almost certainly noticed that I've had some connection issues for whatever reason for the past week or so, that I have no idea where they're coming from. But I reset my modem to factory settings, and hopefully that solves it. I've been testing my connection for pretty much the entire day, and so far it's kind of holding up, as in I was able to stream to YouTube for a good 2.5, 3 hours without it dropping off. So I'm hoping I've solved it. I also tweaked some settings, also in the hopes that, I solve the connection issues. But if I still have connection issues, I'm pretty much just going to request a new modem from my ISP. But on today's stream, I'm actually pretty excited about it, or actually very excited about it, because it's planned content, and it's planned content with another content creator, an actual expert in the field of the research that we're going to talk about, which is some neuroscience research, some optogenetics research, which is great because for a number of reasons. One, I cover neuroscience research from time to time and psychological research, and I am definitely not an expert on neuroscience or psychology, and so it's great to have an actual expert to provide, actual expertise as opposed to me. Maybe correct anything that I say that is wrong or inaccurate, and yeah, pretty much just to call me out on any misinformation that I might be spreading, because I definitely don't want to do that. And I always try, at least, to be transparent when I'm speaking out of turn or out of my area of expertise, but it's also good to actually have someone who's actually qualified to do it. But yeah, and also next week, I should have some other content planned, but nothing's really confirmed yet. At the very least, I should be doing some science and research streams, again, assuming I don't have any other major connection issues, but that remains to be seen. But pretty much we're going to get right into it. So what I had planned for this stream is to pretty much go over one specific paper, a neuroscience paper, specifically an optogenetics paper. And I've covered I've given a brief primer about optogenetics previously, which you can find the VOD on the YouTube channel if you want. And now we're going to actually dive into pretty much a pure optogenetics paper, research paper, that tries to understand behavior at the level of the brain using a rodent model. But Neurolicia will certainly provide way more insight than I can. But I think it'd also be, as mentioned, I chose optogenetics because I think it's a great intersection between neuroscience, biology, biochemistry, obviously psychology as well, and a whole host of other disciplines. So while I obviously can't cover all of them, but I can maybe provide a little bit more insight onto the biology/biochemistry, and they can definitely provide way more insight onto the actual neuroscience and psychology, and the whole behavioral aspect that they're trying to understand with a research like this. So I think we're going to just get right into it. because I'm super excited and don't want to waste any more time. Message them. Have planned this out previously. Hello, hello.
Hello, hello.
All right, gonna get things set up on my end.
Yeah, no worries, no rush.
All right, hopefully you can see.
I can see you, yes. I assume you're able to see me and my lovely dog in the background
Yes,
here.
absolutely. Perfect. Get things more organized and lightly.
Yeah, I'm gonna make sure I have my paper up and everything like that here, so I can see my copy, but I'm probably just gonna look at the screen on yours because that'll be easier. But then if I need to like scroll to something else, you know, know how sometimes looking at something while you're talking about it and being able to kind of bebop your mouse around. Anyways.
Yeah, I know exactly what
I have my notes on there too. I didn't take extensive notes. I kind of was like, oh, I'll probably remember most of this.
I just highlighted some stuff. I mean, I took some like very casual notes in Notepad, but
nice.
Nothing. Definitely, they barely pass as notes. Put it that way. Second.
Making noise over here. Knocked things over trying to get my water bottle. No worries. Maybe this is a good time for me to kind of like pseudo introduce myself.
Yes, that is how things should probably work. So yeah, introduce yourself. You know, if you want to give any, pretty much whatever you want to do, go for it.
Sure, yeah. So I, like Corey, am a SciComm streamer. My stream is a little bit more chaotic. I tend to get a lot off topic. Corey's really good at staying on topic, and I absolutely love listening to what he's got to say. So I'm really excited to be here. I'm also a PhD doctoral candidate, but in neuroscience and psychology. And I also have a master's in experimental psychology. All of my work previously has been in rodent models. I've worked with rats, I've worked with mice. I currently work with mice, looking at pain models. But my master's and my undergraduate theses were both looking at addiction. And so I think I have at least a decent amount of expertise to be able to speak on this particular paper. Not to mention that I also know the authors personally. They're fantastic scientists, fantastic people. You know, inside and outside of science, I highly respect this group. So I was excited when Corey picked this paper. I kind of gave him a suggestion of a couple groups to look at, and this was one of the short list. So I'm glad we went with it.
Yeah, so do you want to maybe give some, like, so you mentioned that you know the only reveal what you want to reveal, but like as in like, have you, so from what I recall, or it's in the paper, University of Michigan, as in like, have you like been like a visiting researcher there, or been to the university?
I've not. So Mike I met at a Society for Neuroscience conference, which is our kind of like disciplinary big conference, but like 20 to 40,000 people every year. And I met Mike there. I met Shelly at a different conference that was in Greece, so that was amazing. But yeah, it's always kind of been interdisciplinary, and then I had the great privilege to meet Kent. He came and was a speaker when I was in my master's degree at a previous institution. And I got the absolute pleasure of sitting down to dinner with him and a group of other researchers there, and being able to kind of talk face to face with him one-on-one, which was a delight. So yeah, Mike, I talk to Mike frequently. Shelly just had a baby. They're all wonderful people, so yeah.
Okay, I'm still like adjusting some minor
No
things
worries.
on my.
So, in the world of addiction neuroscience, at the very least, Michigan is one of the places that's kind of like a mecca, if you will. So there's a couple of institutions that, outside of just general prestige, like the MITs and the Stanfords, those are prestige universities, no matter the discipline. But then when you go into kind of a sub-discipline, so addiction neuroscience, there's a couple of universities that otherwise aren't super remarkable to a layperson. And Michigan is one of them. They house a really good, specifically addiction neuroscience group, as well as Lethbridge, Alberta has another pretty decent little neuroscience group there, but it's a bit more broad neuroscience.
Okay.
So, yeah.
So yeah, I have the paper up on my stream. Should be like hopefully clear. So actually, it's probably better if I actually also share it in the Discord so that pretty much you see what I'm seeing.
Yeah, that would be useful so that I make sure that I'm not talking about a different figure from you and we don't have any kind of miscommunications there.
Yeah,
But as
exactly.
you wish,
I
for
will do
sure.
that.
For those of you wondering, my little black spot in the background is my dog. She's very tired. I took her on 2 long walks today. So, all right, I'll just plug in here to the stream. Excellent. Okay, I've got it up on my end, so that's perfect.
All right.
No sweat. No sweat. So Mike is actually he is a professor at Concordia and also works with some researchers at University of Montreal with a lady named Anna Noel Samaha, who's also an addiction researcher and uses optogenetics and self-administration models similar to what we're going to discuss here. So Mike is actively in this area. Shelley is out in University of South California, something. So she's in California at the very least. And they're both Dr. Mike Robinson, Dr. Shelley Warlow, and of course Dr. Ken Barrage. So
Gotcha.
very well in their fields.
1 second. Oh, the wonders of technology. I am going to figure it out. It's just
I am just impressed that you can figure this out. Like, I was gonna try and have people come on my stream, and I'm like intimidated by it. So you're already doing way better than I would.
Because like yeah, I had it, but then like me sharing the screen sort of like messes up the camera.
Oh, does it?
1 second.
You don't have to if it's gonna be a huge issue. Because I do have the paper up here. I just
yeah, but well, I think it's better to have well, as opposed to 2 invisible heads speaking. At least I am going to figure it out. 1 second.
Yeah, absolutely. Absolutely. So I know I was there, I think, in one of your streams where you did the little primer on optogenetics, and I picked up actually quite a bit of information just in terms of the like more molecular kind of protein level, if I'm not mistaken, that you're talking about, and what exactly specifically like fluorescence and stuff like that is being driven there. So that's something that I'm super excited about, being able to hear what you've got to say, because as much as I'm like the expert in the room when it comes to this specific very niche kind of subtopic, I think that there's definitely a bi-directional sort of information passage we're going to have here. So
and actually, so I think maybe a good starting point, because this is a paper about addiction, and as we sort of like touched upon, like this paper appears to be working with a specific model of addiction, or maybe like one of the models of addiction. Do you maybe want to cover, well, obviously you don't give a lecture of all the models of addiction, but maybe like the most, I don't know obviously how it works since I'm not in the field, but are there like, sort of like 2 mainstream ones, and then like lower is like a hierarchy?
Yeah, so well, it really depends. It's like a lot of places in science. There's contention. There's camps that are somewhat in opposition to each other and kind of rivals, if you will, if you want to make it sound a lot more interesting than it really is. But they all, of course, respect each other greatly and sometimes even work in each other's favor and everything. But suffice it to say, there are several different theories of addiction. The one that this paper and this group subscribes to is called the incentive sensitization model of addiction. And in order to understand that, really, it kind of necessitates that we talk a tiny bit about other theories of addiction, which there are basically three kind of historical alternate views of addiction. And I want to just like preamble say that none of these are necessarily all right or all wrong. It's just some of them aren't sufficient to cover all of the aspects of what we consider to be addiction as separate from just casual use of addictive substances or behaviors. So the first one is what is called the allostatic theory of addiction. And that's just a jargony word for basically your internal regulation. You have like a whole bunch of levels that you want to keep in your body. You want to keep your temperature correct, you want to keep your hydration, etc. All of that sort of stuff you want to keep within a tolerable range, otherwise you die. So that theory of addiction kind of focuses on how taking drugs, I'm going to use drugs, but this could apply to a lot of different addictive behaviors and pursuits such as food, but drugs is the easiest one to understand. So initial drug taking is because, ooh, you like it, it feels good. And then what happens is when that drug is on board, it causes changes in your body. And in order to keep your body at homeostasis, to keep it within that realm, your body builds tolerance to the drugs. So you need more of the drugs to get the same effects, and your body adjusts, and then thus you have to keep taking the drugs. Otherwise, you'll go into withdrawal. That's kind of their theory of why people become addicted and stay addicted. The next one is called the habitual compulsion theory of addiction. And that basically just suggests that behaviors, addictive behaviors like smoking a cigarette, drinking a beer, injecting drugs, etc., are overtrained behaviors that were initially goal-oriented, I want to get high, but then become overtrained and, transfer from being something that's purposeful to being something that's kind of impulsive. And habitual, much like, when you move places or you change jobs and you accidentally take the wrong turn, or you put the keys in what it used to be the spot, or if you're on vacation, you wake up and you're like, for a second, you think you're still in your bedroom, but you're not. That's the sort of brain mechanism that this theory would be relying on. And then, of course, the last one, before the incentive sensitization theory, is the pleasure theory of addiction, which is basically that they feel good and we want to feel good, so we keep taking them. So all of these, they have truthiness to them. They we can relate to those. But a lot of them are insufficient. So many, if you speak to addicted individuals, lots of them will report that they don't even actually like their drug of choice anymore. Most smokers, they're like, I don't love cigarettes, I just do it because I get irritable if I don't, which would adhere to that first theory, right? But also, people relapse years after withdrawal effects go away, so that can't completely explain it, right? So the incentive sensitization theory, in a very small nutshell, is that addiction is a disease of aberrant wanting or aberrant desire and craving, wherein drugs and their associated cues become super kind of motivational magnets. So a really kind of relatable example in North America, at least, is if you're driving home and you're like, oh yeah, I'm going to go home and have dinner like I usually do, but you drive past a McDonald's and you see the golden arches, a cue associated with that food, and you get a craving for a Big Mac and you just have to turn and go in. Or, that's a food related one. Maybe, you wake up in the morning and, you see your pack of cigarettes in your lighter, and that triggers you to want your cigarette at your usual time. But maybe there's a different time that you wouldn't usually smoke, but you see that and it instigates that craving. So that's kind of the really, really quick and dirty version of all of those. And I personally am more of an adherent to the last one. the incentive sensitization theory. It's quite a mouthful. That theory, interestingly enough, is the product of Kent Berridge's work in collaboration with another researcher at Michigan, Terry Robinson. So he was one of the people that pioneered this idea.
Yeah, so those were like really great explanations. And so obviously you're way more knowledgeable about the field than I am. But in terms of like these particular models, you know, is this like a really like split camp in the field? Like, you know, they're really like strong adherents of this one, and they're just like super convinced. Or is it more like more up in the air of like, wow, this is the one we're going with, and we think it's the most true? Basically, you know, is this like a really great source of conflict at conferences? Well, as far as you can tell.
Yeah, so I would say, especially for those of us that I would like to use a sports analogy, if I might. Even if you're not a sports person, you know, football fans, American or European, like soccer. They are diehard, scream at the top of their lungs, wear the jersey, level, invested in their team, in their camp. This is much more like golf, where
Okay.
everybody's kind of like, oh, they're my favorite golfer, but like it's really a gentleman's kind of like, oh, well, what about this? And everybody kind of plays ball and is quite fair and balanced with a couple exceptions, but I've never seen anything get particularly heated in terms of the actual theories themselves. Sometimes the niche stuff and how behavior and results are explained gets a little bit dug at, and that tends to play out more in the review process. But yes, there are camps, but you know, it's all white guys in tweed suits being pretty nice to each other and just being like, well, have you thought about this? You know,
Yeah.
so
it's not you know, impassioned, you know, fights or brawls at the conferences. It's more like, you know, some friendly debate, and everyone, you know, no one's, at least is, correct me if I'm wrong, but hopefully no one's trying to like break someone's career, or like, you know, tear someone down. But I also know it's academia, and that is totally a thing, unfortunately.
not the major players. So I think it would be fair to say that people that are like that don't tend to make it super high in the field and stay there in this day and age. And this is a pretty rapidly evolving field. So even though these are, you know, boomer generation individuals mainly doing the theoretical groundwork, you know, 20, 30, 50 years ago, a lot of the active people doing experiments and stuff like that are young and eager to kind of integrate things, and also are seeing a lot of evidence to suggest that it's not as cut and dry. So it would be kind of obtuse for any self-proclaimed critical thinking scientist to be like, that's wrong, because it's just not that black and white. So any sensible scientist isn't gonna, you know, scream down someone's neck about it. Everyone's quite fair and kind.
Great. Perfect. So I think we set the stage more or less in terms of like the overall like macro scale, you know, topic of the paper. Pretty much, you know, about addiction, and maybe trying to tease out how addictions form, or maybe how like habitual behaviors are formed. And, yeah, go ahead.
I was just gonna say, so I would be really careful to the nomenclature here. Using the word habit is something that it works really in lay language. People talk about kicking the habit and stuff like that, and that tends to be a relic of one of the previous more historical views of addiction. And within the context of this paper, that's a very kind of meaning-laden word. So I'm going to avoid using it, but just so that the audience knows, kind of thing, that it's not so much that it's wrong, it's that this group would very much not use that word because it's laden with meaning.
Yeah, for
right?
sure. So that is a good thing, because like I'm aware of how, let's say, getting your terms right is really important in science. Even just something as simple of like, if you don't know the difference between, you know, at least for me, if I didn't know the difference between precision and accuracy, I would totally
Absolutely
be questioned on that.
That's a great example.
So like, yeah, if I use the term habitual, but it's not actually, you know, a meaningful term either in your field or when referring to this research, like definitely like let me know. So like, it really is like in the vein of like addiction. or maybe trying to tease out the early development of addiction. Would that be a more appropriate way to phrase it?
Sure. I mean, I think what they're getting at here is maybe one of the mechanisms that's driving what they consider to be one of the major factors in the beginning stages of addiction, like you were just mentioning, and the maintenance and progression of it, which is craving. So, I
Right.
mean, that's kind of, if you wanted to take a central word and make that the thing, it would be wanting and craving, which is primarily what seems to be the driver from this perspective.
Oh, so maybe like sort of early development, potentially on like the pathway to like addiction or something, maybe
Yeah.
pathological.
So there are groups that actually explicitly do that research. I've published research on how early life experiences contribute to addiction and maybe the early development of addiction, in terms of like how the sequence of events typically goes when we look at addiction, which isn't really present here in the paper, but it certainly is related research. I would say that this is more related to the transition from the beginning stages of addiction to that narrow, intense focus when people, that's all they can think about. If you think about really stereotypical addictive individuals and how they're like focused on getting their fix, I think that's really what this is trying to model in this specific paper.
Right. And so then, yeah, to sort of, again, further set the stage. So for this paper, they're looking at a very specific region of the brain, kind of like narrowing in, as sort of highlighted in the title, central amygdala and their specific region. So do you maybe want to, provide some relevance in terms of these particular regions of the brain, or rather they sort of make the distinction of like the CEA versus the BLA? Um, yeah.
Certainly, yeah. So to just kind of contextualize people that aren't even familiar with any brain anatomy, there's different regions of the brain that are like, cortical regions that tend to be human specific, and then there's more kind of primitive regions. One of those primitive regions is the amygdala, which if you have taken usually any introductory psychology or neuroscience course, or even done a bit of light reading, you tend to see it associated with fear. And that's true. It's very involved in fear responses, but as is the case with many things in the body, particularly in the brain, the amygdala is much more nuanced than that. So it's not that that's wrong to say, it's that on top of fear, which is very involved here, it's also about what we call valence. So this is the goodness or badness of something. So as much as like, chocolate is sweet, it's also, holy crap, seawater is really salty and aversive. So both of those things are going to be encoded in the amygdala. And the differences between parts of the amygdala, the central part, which is closer to the center midline of the brain, or the basement, bottom, lateral, so the bottom side of the amygdala, in so much as they're different, is it might be a little bit, I guess, mincing kind of thing for the purposes of like a general understanding, but the paper does get at something here that I think is really important, and that is that the central amygdala maybe seems to have some connections with motivationally relevant areas of the brain that are tied to addiction, and it tends to be an inhibitory area. So what we think might be the case is that specifically that central part is involved in taking the brakes off of certain areas of the brain that are involved in addiction, and that the basolateral amygdala is not.
Okay.
It's involved in a whole bunch of other stuff, but that's
Sure.
outside of the realm of the paper, right?
So if I can maybe clarify, or maybe just maybe for my own conceptual understanding, because you use the term like taking the brakes off. So is it really more like taking the brakes off, or is it like, would it be also apt to say like putting your foot down on the gas, or both, or really
So
is like
that's
just
a very good question. That's a very good question. That's a fine distinction. So this is something in neuroscience that we call disinhibition. So usually there's an inhibitory signal that's driven by GABA, which is basically is telling certain circuits to turn off. or slow down or don't fire as much. And there's also something driving that GABA signal. So you can think of the GABA signal itself as the brakes on, say, the dopamine system, which is involved in reward. And if you inhibit that signal from the central amygdala to those brakes, if you inhibit the brakes, then you're going to allow things to go forward. So it's a very fine distinction, and I'm really glad that you asked that, because it's not the same as excitation. Excitation is driven by a completely different neurotransmitter called glutamate, which is an excitatory neurotransmitter that is involved in some of these processes, but not specifically the ones that are addressed in the paper.
Okay, yeah. So I did want, because I know that there's a distinction, but I want to make sure
Mhm.
that, like, yeah, well, also that I sort of have it hopefully accurately conceptualized in my mind. So yeah, that's good to know. So I think we set the stage more or less of like the overall macro scale of the entirety. Well, if you feel like we've left something out, or I didn't fully provide enough feedback, by all means, feel free to interrupt me.
Um,
Yeah, go ahead.
I think it might just be useful for me to like really simplify incentive motivation.
Sure.
That's just a fancy way to say craving. So when you look at this title, it kind of feels really jargony and in your face. But if you had to, make it into like a teach it to me like I'm five title, it would be laser control of the amygdala, turning the amygdala on enhances craving for a specific thing. And in an application in real life, that thing would be drug. Here, it's sucrose, but
Yeah,
yeah.
or salt, as we'll find out shortly.
Yes, yes, exactly.
So yeah, so thank you very much for providing that context.
Mhm.
Um, so I think we probably maybe just start with, so obviously I don't think it would be appropriate or even a good thing to like dissect the methodology, but just a broad overview in terms of like, so like they're pretty much using the animal model, the rat.
Mhm.
Uh, so uh, in as sort of we touched upon earlier, in the optogenetics research that I sort of like casually follow or have read, like they most often use mice, and here they're using rats. Is that like relevant, or it's just like since the Barrage lab uses rats, and they just is it more like tradition, or is there something like specific about rats that's like special here?
There's a couple of explanations here. One of them you very much nailed. You can't do both because mice are predated upon by rats naturally. So if you were to do mouse research in a place that's also doing rat research, you would be self-confounding because the mice would be afraid of the smell of the rat. And also the rats would be like, oh, smell food, yum, and distracted for that reason. So that certainly is one reason they're a rat lab. The other reason that using rats is beneficial is they're just frankly easier to train. They're just smarter. They're like a little bit just less prey driven, so they're less afraid to explore and interact with things. They're bigger, so if you wanted to do, which this lab and related labs also do, self administration of a drug using an intravenous model, mice are way smaller. That's way harder to do. When things get
Right.
small, things get hard, right? So bigger is usually easier because you don't need scopes, you don't need as tiny a tools and as firm or as steady of a hand. But both are fine. Mice, the reason that most people use mice is because they're cheaper, and because you can do a little bit more of genetic modifications in them, so you can do kind of some more elegant designs and stuff like that. But of course there's a trade-off.
And in terms of like the actual, if you're able to say, like in terms of the actual like neuroscience, as in like, you know, basically is all the circuitry that we're observing here in the rat that they're, let's say, using and another research, is that all like, so I basically don't really know the actual difference, aside from like the obvious of like size, and
Yeah.
people view rats as whatever more disgusting. But in terms of like at the level of the brain, do you happen to know if like there's any like major differences? As in like, could you basically like replicate this, or like get similar results from mice with this? It's just like the technical challenges that are the issue.
That's also a really great question. You could definitely do this type of a model in a mouse very easily. Specifically, this one, optogenetics, is very easy to do in a mouse. The only thing is that the size of the actual cap, where the optrodes go in, is going to be basically the same size. We don't change the actual optrode size between mice and rats. It's the same one. You just have less surface area to work with. But people do this all the time. But in terms of their brains actually being similar or dissimilar, from my kind of like gross anatomy view of it, it's basically kind of just a scaled up thing. And in so much as a chimpanzee is a scaled down version of us, right? Like,
Right.
so we all have fear responses: humans, dogs, cats, rats, mice, you name it. Even flies have fear responses. But consider that the reason that you're being afraid really colors the situation. I'm afraid of failing my PhD. I don't think a mouse has that concern. So even when we're talking about the same circuitry and the same behaviors, they might not be kind of colored the same way. And this is something that's really hard to prove or disprove. Mice aren't little rats. Rats aren't little people, but they're the best that we've got. And they're similar enough for these purposes, is what most people conclude.
Yeah, or at least trying to tease out maybe some like rudimentary circuitry that maybe is presumably, though I think the working hypothesis is that like it's conserved in us, maybe not to the exact same level,
Yeah,
but like totally there's information to extract from results like this that should be hopefully applicable.
yeah,
So
that's
yeah,
a great answer.
yeah, I think that answers sort of because that was actually one of the things that really like stood out to me. Like yeah,
Mhm.
wow, why rats? Which
Rats
uh,
are pocket puppies. They're way easier to work with, and mice are so stinky.
that's
Having worked with both, I absolutely love rats.
Yeah,
Mice
that
are
that
okay.
I definitely know from your stream, which is super interesting. Without getting like too sidetracked, but in terms of like
Yeah,
public perception, right? Like most people like the rats away from me. Not that everyone's gung ho about mice, but definitely
yeah,
more people are would be willing to hold the mouse or like keep a mouse as a pet as a rat.
yeah, yeah. That's definitely true.
But it's I had no idea that basically they were essentially like more tame, and like overall I guess. Well, so
Uh,
this also springs to mind again without getting too sidetracked. But you mentioned earlier that rats are like more willing to explore, and I'm curious. I understand this probably may or may not be like your area of expertise, but like so they also use like mice and rats for like depression research and sort of like the open field tests and stuff like that. So like I sort of is that like a potential like confounding factor? Like well, in terms of willingness to explore, like basically are rats less
yeah.
amenable to like an open field test than mice? Because they're just gonna
No, you can use an open field test in mice and rats. I've done it in both and published in rats using an open field test. The nice thing about these tests is even if there's differences between species for whatever reason there is, you basically are doing a pre and post, a within subjects baseline. So
sure, yeah.
you
Sure.
you know you're measuring the mice or the rats against other mice that have or haven't had the previous thing, or sorry, the intervention that you're wanting to see if that changes presumed anxiety behavior, which is what the open field is supposed to measure. But it doesn't really matter if rats and mice are the same because in your experiment you would always be looking at between mice and mice
Yeah,
in
the
different
relative
conditions.
change of like this rat who's whatever happy versus depressed and how they respond to that open field test. And yeah, I mean that makes total sense. For sure. I just thought like maybe since that they're as you their willingness to explore is just like greater, or like maybe it's just not tweakable and so they're not amenable to the open field
Yeah,
test.
so
But
you scale it up basically because an open field to a mouse is about a foot by a foot
right.
in a lot of cases. For rats, I could probably have a comfortable nap in an open field for a rat. Maybe not. I'm pretty tall, so maybe not a comfortable nap, but a nap nonetheless.
So yeah, they're modifying these rats. They're in many ways, both genetically and surgically. So
Mhm,
without getting into like the nitty gritty details of like the surgery, but they're basically, from what I understand, and definitely correct me if I'm wrong, but
mhm.
they're injecting essentially genetic information in certain regions of the brain, the regions that they're interested in that you sort of described earlier, the CLA, rather the BLA and the CEA.
That's right.
Is that correct?
Yeah, so we take a viral vector, and this is one of the few good things that came out of COVID-19 is lots of people know what spike proteins are. The outside of a virus, and this might be somewhere where you can step in and tell me how correct or incorrect I am, but I basically, in a really kind of pop psychology sort of way, describe it as they take the shell of the virus, they take all the goop that makes it, in this case, an adeno-associated virus, which is the virus for kennel cough, if anyone's a pet person. AAV is kennel cough. And they take the stuff that makes it kennel cough out, and they put the stuff that expresses this light sensitive channel in, and then it goes and it expresses just as effectively as if it were still kennel cough, but instead of making that happen, it expresses these proteins wherever you end up putting it, and it proliferates in that area. So an interesting thing that I think gets kind of breezed past for lots of people is you have to wait. You do the surgery, you implant the leads and the virus, and you have to wait anywhere from 2 to 6 weeks, sometimes up to 8 weeks, for the virus to transfect, is
Right.
what the word is. And if you do it way too past that, then the virus over transfects and goes into areas that you don't want it to go into.
Right. So that's exactly because when I was reading this, of like, I don't know if you can like see it. I have it highlighted sort of like here, of like they're literally injecting a certain volume, 2 microliters,
Yes.
like into presumably the region of the brain. So like I was just like curious, obviously, since I've never done this, and I have no experience with rats or mice, but like are they just like kind of like hoping that they're getting the right regions, or is this like some sort of like guided injection, or like it's sort of the presumption that if they wait long enough, the CEA and the BLA will be transfected, as you say, but if wait too long and like the whole brain would be transfected, and you can do well if you use the same light, you're just gonna light up the whole thing, presumably.
Yeah. So that's a really great thing, I think, to draw attention to. And the answer is we certainly use a very precise technique and very precise equipment. There's something called a stereotax, which is basically a device that has arms on it that you can control up, down in the Y plane, left, right in the X plane, and wait, I'm getting this wrong. The Z plane. In every direction you can control it. And basically there's also been a lot of histology and brain staining and stuff done on different sections of mouse brains and rat brains at volume, so that you can kind of get the net average of where it would usually be relative to top or bottom, much like you would do in like geological research. Like if you want to get to a certain layer of things, you need to go this deep to find that area, but you can't go this deep over here to get to that area because it's underneath here. So you need to kind of go according to, it is literally called a brain atlas. And then you take a very tiny needle and you go through the tissue that's before that area. And then once you get to that area, you squirt the very small amount of liquid out, and then you slowly pull up the needle so that you don't create a vacuum, and suck it all back up.
Right. And I'm curious if you're able to say, so obviously, in rodents, so, let's say the approximate size of like the rat, is like size of a pea or whatever, a grape, or
The size of the brain?
yeah, the actual, I'm trying to like, yeah.
A rat brain would be about this big. It would be about the size of a walnut.
Okay. That's actually much bigger than I anticipated.
Yeah, and a mouse brain is probably, I'm actually going to bring one on my stream eventually. I just keep forgetting to bring one from work. It's probably, I'm trying to find something. It's bigger than my thumbnail.
Okay.
But it's probably maybe a centimeter-ish deep, top to bottom, and centimeter or two long.
So essentially, like less than half the size of like that walnut rat brain, if I'm understanding.
Maybe like a green grape.
Okay, yeah, okay,
I
gotcha.
don't know why I'm using food things, but yeah, so
Well, no,
somewhere around that.
I think that's
Depending on the mouse too, because there's different strains of mice, there's different strains of rats, right? So
And so, again, correct me if I'm wrong, but so like they're injecting that small liquid, and they're targeting certain areas of the brain, or they're attempting to target a certain area of the brain. But as far as I know, again, correct me, but like these aren't like defined borders of like the border between the U.S. and Canada. Of like they're presumably not getting like every single cell, or like it's not specifically defined like, oh, this collection of these cells is like specifically the CEA or the BLA. It's just kind of like, is it possible that maybe half of the CEAs or 3/4 is transfected and like similar to the BLA, or like they're confident that they're getting all of it?
So they would, of course, confirm with slicing the brain and doing imaging afterwards with those fluorescent proteins that are tagged onto this, like when it says AAVCAG, blah, blah, blah. The GFP part at the end, which I think you can speak to with more expertise than myself, stands for green fluorescent protein. So it makes it glow like a glow stick when you turn a certain light filter on in a microscope. So you can actually see, if you got in the right area, and if it overexpressed, underexpressed, if your needle was clogged and there's nothing there, etc. Specifically, the question that you or the point you raised about which populations of cells in that area maybe got infected, we have to remember that it's not just one road. These are like interconnected highways in your brain that go to multiple places. So in this case, they very much did not do projection specific, because that's actually hard to do in rats, which is why a lot of people use mice. You have to use a model called a Cree-dependent model. So you make it so
the Cree-log system, if I remember.
Exactly. Yeah. So then it only goes to cells that have basically that flag that says come express here. Yeah. But the mouse itself, or the rat itself, has to be expressing that already, which is a whole different ball game in terms of making that happen. And then you have to use a Cree-dependent virus, which is more expensive, etc. But that is possible. They didn't do it here.
Okay, yeah, because that sort of, so I'm most familiar with the Cree-log systems. That's more of like, whatever. This is a genetic technique, but this is more like really genetic technique. So that also like sort of stood out to me. Like, oh, they're using sort of a different system here, which obviously it seems like it's way more convenient. So just like whatever, inject.
Yeah.
And do you know if you're able to say, since again, I have no idea in terms of how complicated it is to actually inject into a brain, but or at least a rodent brain, and where these areas are located. Like, do you know if they're let's say easily accessible? Or I guess my overall question is like, is there potential for like damaging structures as you're doing it that you might not be aware of, that might be producing something?
Yes, of course. You create damage on the way in. It's like putting a really tiny straw in there. So you kind of are going to be damaging all of the brain down to where you are. And these are, like I said, they're older brain structures that are less changeable than cortical structures that are more at the surface. So they're deeper. They're closer to the center part of your brain, the core of the apple, so to speak. So yeah, you have to go through a lot of brain tissue to get to those areas, and you certainly damage along the way. The way that we control for whether or not that's what's driving the behavior is when we use a control virus, right? Because they still get the surgery. It's much like what we would call a sham surgery. So in order to make sure that it isn't having had the surgery that's causing the effect, rather than us driving that specific population of neurons or that specific brain region, we say basically, you've had the surgery, you've both had the surgery, just you got juice and you got not actual active juice.
Right.
And I say juice because it's just a lot more relatable, but of course it's virus, right?
So like presumably, the sham surgery is like sort of to maybe correct or to ensure that, oh, okay, well, this brain should be damaged in a similar way, hopefully identical way, as the actual ones that are getting whatever the treatment, so to speak. Okay, yeah, that makes sense.
Yes, that's a very succinct. Yes.
Second. Okay, so I think that covers sort of like what they're doing, how they're modifying these rodents, and then so yeah, the genetics part, or rather the genetics part of the optogenetics, And then like yeah, the opto, the laser. So like then they're surgically inserting those lasers to those regions, right?
So a fine distinction here. They're inserting a optrode. The laser is external. It's quite a big thing. It's
True,
really
yeah, yeah, yeah.
powerful.
It's
And then
transmitted, yeah.
we plug into that. So they're not walking around with wires out of their head or an actual laser emitting diode or anything like that on their head. They're walking around with leads that you plug into when they're doing the thing. So, they have that really cool little picture or still frame from the video that demonstrates, what that actually looks like on the rat's head. And yeah, so you cement those in place using actually dental cement, what they use in dentistry.
Okay. Interesting. And I think they mentioned it here. So these are like very small, 200 micrometers or microns. Yeah. So
microns. Yeah.
So this is essentially like roughly or almost exactly a fifth of a millimeter, if I remember my units correctly.
I'll trust your math.
Yeah, because 1,000 microns should be 1 millimeter. So 200, that's 1/5. So like yeah, pretty much. I don't know how that compares to the a human hair, but I think pretty much it's like roughly on that scale.
I'd say it's a little bit thicker. The ones I've used, I don't know if they're exactly the same. I don't have, all of my measurements of my optodes that I've used historically memorized, but they're likely very similar to the ones that I've used. And the thing is that the thinner you make them, the less damage they make, but also they're made out of glass, right? So they're very fragile. And you have to polish the end so that the light travels and doesn't scatter like it would in like a prism sort of situation, right? So it's quite a refined process to make the optrodes, and it's trying, to say the very least.
Yeah, like I can only like I said, I have a rough idea of 200 microns, and like yeah, that's like pretty close to like really not actual paper thin, but like these are like frail things, and these rodents, they want to move around. I have to imagine that these are like pretty flexible, but I'm sure like
they're brittle as heck. They're 0% flexible. If you drop it, it's gone. If the rat or whatever catches its head on something, or another rat, if you have them pair caged, chances are, if you didn't cement well, you put actually little screws in, and then you build cement around it so that it doesn't just like pop off. But yeah, it's the attrition rate, if you're not a good surgeon, it can be substantial. I would say like out of 10, you might lose 1 or 2 on average, kind of thing, if you were a beginner at it. But then you would use the mouse or rat for something else.
Yeah, I was gonna say, so like in that case of like the optical fibers break, is it like, can they be like reinserted, or like this mouse is like disqualified, like pretty much that's it, we're done, we're gonna start with a fresh mouse, fresh surgery, fresh optical fiber? I'm assuming that that's probably the approach.
I mean, that being said, this lab does a ton of opto. I highly doubt that they lose any to that sort of situation, unless it would be a far outlier to a lab that does this regularly, because they have it down to a fine-tuned process. But when I was learning, it's brain surgery,
yeah,
and cementing things into a brain of someone that's walking around. So it's not easy.
Yeah, the expression "it's not brain surgery" does not apply here. Literally is. And so I'm curious if you're able to say, this is just like my other, I guess, maybe animal welfare side of like, so like, yeah, if like the optical fiber breaks and like this mouse is no longer qualified, is it, are you able to see if it's just like sacrificed or become someone else's pet, or?
We, of course, keep their brains, so it does not become someone else's pet. We want to extract as much information out of animals that give their lives to science as possible. It would be utterly irresponsible to just be like, oh, sorry, mouse, go live your life, or sorry, rat, go live your life now. We want those tissues. We want to learn as much as humanly possible, or ratly possible, I suppose, about those animals. So you can still see, was your electrode in the right place? Because they do these experiments, specifically in this paper, they do them over days and days, right? So if they did 20 out of 21 days, and then they had an electrode issue or something else, because that is quite rare, you would still use the first 20 days of their data. You would just get rid of the data after. And then you would
Okay.
still want to look at their brain and confirm that you were in the right place for those 20 days, right? Because not just, for practical data reasons, but for humane reasons.
Oh yeah, so that's actually something I didn't really like consider. I thought maybe more in the process of like surgery in early days, that maybe something goes wrong with the optical fiber. But I guess it's also for sure, as you just described, potentially possible that like, this rat might be like 90% done all of the studies, or the experiments that you want to run, and then like at the end, like close to the finish line, like it's something goes wrong, and then yeah, the rest of the data collection, you can't collect basically. Guess that presumably does occasionally happen, but probably not with this lab. They're super experienced. Yeah, this is like really, really tricky experiments, science to conduct, where like pretty much you have multiple failure points, basically. That's the main message.
Absolutely. Yeah, definitely.
So I think that more or less covers what they're doing to the rats.
Hmm.
And well, I guess maybe the last thing maybe I don't know how useful it is, but this is again another thing that kind of like stood out to me, is like, well, with optogenetics, the opto part, light, they're not just using like your standard light bulb, or my LED light that I'm using now, or my ring light, but like they're using a very specific like wavelength of light. Right here, they're using like I think blue, as
Yes.
highlighted in the figure.
Yes.
So like, and that's presumably that's relevant, right?
Yeah, so it's basically what the channel responds to, and I will caveat this with I'm not entirely sure, but I'm relatively confident that this is correct. When they developed this, they developed it based on I think genetic material from blue-green algae or some sort of algae that blooms in light. And of course, it only does that for sunlight. It doesn't do that for moonlight. It doesn't do that for, that sort of thing. It's specific to a certain wavelength of light because that's what's adaptive. So you have to give it that specific type of light for it to activate. And of course, there's ones that will activate to yellow light that are usually inhibitory, and kind of various kind of forms in between. But blue and yellow are the most commonly used ones. And it does have to be that specific wavelength of light, the specific, I can't remember what the units are for the life of me right now.
Nanometers, if I remember
Nanometers,
correct. Yeah.
there we go. And then of course you can adjust the frequency and all of that sort of stuff, as well, but it does have to be that color, otherwise it will not work.
Yeah,
And that's the most expensive part of this is the laser.
okay, yeah, well, I guess that's sort of I'm sure, unfortunately, I guess rats are probably super cheap. Maybe not like the surgery and all, like the manual labor to modify them. But yeah, that makes total sense. And this sort of, I guess, my follow-up is like, so I know, as you just described, that the blue light is important, and using that specifically because the channel rhodopsin, it responds to that, but also at the same time, as I sort of maybe covered in some past research on my own stream of like, well, there might be some issues with blue light, or like penetration into the tissues, let's say, is important. Basically, I guess my overall question is like, is it possible that like you're shining this blue light, but it's not actually reaching the areas that you're interested in, of like you have this the CEA and the BLA modified to hopefully respond to light, but then presumably like the blue light is sufficient enough or bright enough that it should reach all of the required regions. My understanding, right?
There's a lot of stops along the way to ensure that, and checks in the post hoc kind of world, that kind of account for this. So first of all, if you're making electrodes, you test them for their efficiency output using a light meter.
Right.
Manually and in a dark room before, and if it doesn't pass a certain threshold, usually like 80 or 90% efficiency, it doesn't go in a brain, because you're not going to waste brain surgery, the actual mouse's life and all of that on a 60% efficiency, because you lose that over time. They tend to get less efficient over time because of scar tissue, etc. So you want to make sure that you're using only the best of the best to go in initially. And then, of course, after, the reason that we keep the mice and we keep their brains and that they can't go on and survive to, do other things in their lives is we confirm visually, you can see exactly where it ended up in the brain because there's physical track marks there
Okay.
in the slice. And then, of course, we use the fluorescent protein that's associated with each of the viruses and the sham virus as well to confirm that it was expressed specifically in that area and not other areas, and that it was expressed, to the right degree. So it's not like just a little bit of expression, and that's when you have microscopy to confirm that all of those things are right. So there's lots of checks and balances along the way.
Yeah, so that's actually something that I sort of noticed, or that's kind of like stood out to me, or like this paper, of like, so yeah, there are checks and balances. But is it fair to say, that the checks and balances like come after like the data collection, pretty much just like confirm, like they dissect the brains, and as you sort of highlighted, oh they're using GFP, so they confirm that okay, we're getting a fluorescent signal where we expect it. That you see that the optical fibers are inserted, but kind of like when you're actually running the experiment, you're essentially largely going on faith that things are working as intended and expected, and it's only afterwards do you answer the question of like, oh, okay, what we did actually like worked.
Yeah, and that's a great point. And I think you're right. There's only so much that you can do without actually like opening it up and looking at the brain, right? But then the nice thing is about science is it tends to build on existing previous science, usually within the group or larger kind of sub area of science. And you know what behaviors you're expecting. So if the mouse is kind of the odd one out and not doing, in this case, lever pressing when they get that stimulation, which is what they hypothesized would happen, you would suspect that they probably have an electrode, or sorry, an optrode issue, or something going on that's extraneous and is a confound kind of situation. But again, I want to stress that that would be relatively rare for a
Right.
specialist. For anybody that's doing this, we don't just like toss a new person in and say, read this chapter on brain surgery. When I teach my students over the course of like a year before they're even allowed to touch the little dial, they watch over my shoulder doing this. So in so much as it would be infrequent, you would be able to tell pretty quickly if something was out of place.
Yeah, so yeah, I guess that makes sense of like, even though it's a fairly new technique, you have, well, since it's new, but it's super popular and it seems like everyone wants to use it.
Yeah.
And so it's like, the research output appears to be like really high. And so it's
This
like,
is the technique. This is it. Yeah, for the
yeah,
last, I think about 20 years. 20 years ago is when it was developed, and then it got really popularized in the mid aughts, early tens, and then now it's, if you want to get into cell nature or science, better have opto or have a whole bunch of other cool cutting edge techniques for neuroscience, at least.
yeah.
Preclinical.
That's sort of what I covered on my stream of like, it was, for Nature magazine, it was like 2015 technique of the year.
Yeah.
Whether that means anything or not, I don't know. But I
I'm
remember.
sure that Carl Deiseroth and Ed Boyden are going to get a Nobel Prize, amongst other people, for the discovery of this technique in our lifetime.
Yeah, that wouldn't surprise me, especially again without getting too, I guess, sidetracked. Of like, it also appears to be, from what I can tell, of like really branching out. It's not just strictly to neuroscience now. I believe it's like even used, or starting to be used more and more in terms of like trying to understand what's happening in like muscle cells and muscle activation. Although I guess that's still technically in the vein of neuroscience, right?
Yeah.
But like, yeah, it seems like it's basically branching out to a whole bunch of other sort of like peripheral fields. I guess still related to neuroscience. But yeah, super cool. So yeah, I think we set the stage, went through overall the macro of the methodology, or at least what they're doing to the rodents, and then I guess now the actual experiments, going
Mhm.
figure by figure. So like,
Sure.
I guess as you alluded to earlier, I can't remember the exact terminology that you used, but like trying to tease out sort of like incentive, or
Mhm.
craving. Um,
Yeah.
and so here they're basically using rodents and sugar, right? Or at least to start off.
Yeah.
Or do you want to maybe sort of you're probably way more equipped to, I guess, describe the experimental design than I am.
Sure. Yeah, so essentially what we're looking at here is they give them the option between two otherwise identical levers, both of which will trigger the delivery of a sucrose pellet, probably banana flavored. That's usually what rats are most enticed by, though you can get
Oh, interesting.
chocolate. Yeah, rats go crazy for bananas. They love it. Banana baby food too. Both of them will trigger, and so the reward itself, the tangible, physical, extrinsic reward, is the same. The only difference is the stimulation of that central amygdala area, the optogenetic stimulation of this area. So presumably the activation of this area is more than the sum of its parts when paired with a sucrose reward. So I think what they show in their results, sorry to give away the farm, is that it's the central amygdala activation in pairing with a natural reward like that really makes it in favor of only responding on that lever, even though they both do the same output. They both have the same results except for the laser stimulation, suggesting that the central amygdala is instrumental in that narrowing and intensity of focus for individuals pursuing something that they're craving, whether it's food, drugs, behaviors, etc.
Yeah, so like they basically have, again, correct me at any time if I say something incorrect, but they sort of have like the two conditions with like, as they sort of in the figure, sucrose without the laser, sucrose with the laser, and then basically, they flash the light and see its effects on, which lever they go to. And then, so I guess my question, or at least the one thing that I'm kind of curious about, if you're able to say, of like, yeah, they're shining the blue light, or the laser, rather. But it seems like they're shining it at a specific time, like 8 seconds, as highlighted in the figure. Is there like a relevance to that? Or is like basically, how is that? Why 8 seconds versus like 7 versus, is this also like historical? I know that like very often in science, it's like, well, this worked in the past and this is what other research did, so we'll repeat it. Is it sort of in that vein?
I can't speak on behalf of the experimenters themselves, but 8 seconds is used in a lot of other protocols in optogenetics in this type of research, but also outside of optogenetics research done in this group using other designs that have nothing to do with optogenetics do tend to use an 8 second sort of timeout window for both of these levers in this situation. You only one lever press triggers one reward, and then you have to wait 8 seconds before it'll do it again. And the reason that that's important, and they mention this in the paper, is that even though the rat learns that lever will only work once every 8 seconds, they have exceptionally good internal timing, they will still go and interact with the thing that triggers the reward. And that's kind of acting like a stand-in cue, like what I was describing earlier with lighters and syringes and other drug paraphernalia. Those are the cues that we would see in real life. This is kind of acting as the cue that's related to the reward, but not, directly. And the fact that they interact with it even during this, sufficient timeout of 8 seconds is really kind of demonstrating that it isn't just that it's making the food taste better or whatever, that it's
Right.
actually instigating this state of craving that's very addiction relevant.
Yeah, it seems, not to give, I guess, everything away, but it seems like, yeah, it's like uh, something is happening at the level of the brain. Well, I guess it's all happening at the level of the brain, but it's not like, oh, this, as you said, it's not like this food tasting better, or like they're getting like more reward from it. It's like through some other mechanism that like I'm actually not even sure how they sort of like describe the mechanism is out. But I guess maybe like we'll get to that. But also whether this is relevant or not. But another thing that stood out to me, it's like, so for each of the conditions, they're like playing a sound. Um, so
Yes.
is that relevant, or is that also maybe like a historical thing, or like maybe just, or actually as part of the conditioning response, or something else?
Yeah, and I didn't get into the nitty gritty of their procedure, so that might be something that they do describe in there. But I believe when they are initially shaping the behavior when they were piloting things out that, associating one lever with one auditory stimulus and the other with a different stimulus is kind of standard in Pavlovian research at the very least, and can be leveraged if you want to examine other things like, fear conditioning. You could see how strong a fear response is to a tone in competition with a different tone that's used in a situation like this. So it's probably a little bit of kind of trying to extract as much information as possible out of these rats, and also a little bit of just kind of, like you said, legacy sort of, this is the way we've always done it, don't fix what isn't broken. And differentiating the two as much as possible is something that I think is important for understanding their relevancy to the addiction thing. Is even very different stimuli to a sound sensitive creature, they do the exact same thing, and it's not that one is cooler than the other. There, you know what I
Right.
mean? Maybe I'm not explaining that super well, but I don't think it's exceptionally relevant
Okay.
to the results here.
Um, and since like they don't really, well, the way they describe it is kind of like whatever, a white noise or something like. So basically, I guess if you're able to say like, is this noise that they're doing, like, do you know if it like startles them, or is it also like, is that maybe part of like what the
Yeah.
intention what they're trying to do, or it really is just like a gentle, like, as they say, tone
So the
kind of
white
thing?
noise is generally used as masking noise, but it'll just go like and it almost never creates a startle response after like the first session.
Yeah,
They
I
become
guess they
very
condition it.
very
Well, yeah,
very very quickly.
that makes sense. And I think
Of
yeah,
course, if you pair it with something negative, they would absolutely startle to it. That's the nature of conditioning. But you're pairing it with something non-aversive here, right?
right,
So
yeah. Exactly. And I do have that example video. I'm gonna see if I can try get it up and also share it. You.
well, I have your stream up on my computer,
Oh,
so
okay.
I can turn the screen on. I have it like blacked out over here, but I can turn it back on so I can see it there. So you don't need to worry about getting it in Discord. Not that I haven't seen it before.
Yeah. Well, it's just a 1 minute 40 seconds video. So I'm gonna play it now. If you, there's probably gonna be a delay in the stream, but I'll leave it up to you now, and I'll let you know when it's done. I mean, not gonna. So they look. I don't know. Obviously, I don't know the typical behavior of a rat. But like in that video, they look very like frenzied. Like is that like? Are you able to say like is that part of like the response, or they're always that frenzied?
Yeah, absolutely. That's part of the response. Certainly, they're going to be mice and rats tend to be very like kind of like,
Right.
especially in newer environments, which these mice or sorry, these rats will have habituated to over time. But then as they habituate to the environment that used to be getting them going really sniffy and excited and explory and kind of jumpy, Now it's the stimulation that's making them juiced to go towards that stimuli, right? That thing that's predictive of their reward, both the physical reward and the rewarding properties of the stimulation of the central amygdala. And you can kind of use that as like a pseudo like analogous behavior to fiending, to use a colloquialism that's associated with human addictive behavior. When people are fiending and jonesing, or whatever you want to call it, when they have acute cravings, it's hard to talk to people. They're like, searching for their cigarettes in their purse. They're like, yeah, I'm listening. You know what I mean? So without projecting anthropomorphism onto these rats, you can see how it's similar, right? So if you just tossed a rat in a box and gave it a chance to get a sucrose pellet just by doing that normal behavior, and there was no opto or anything else like that, you probably wouldn't see such similar effects.
Okay, yeah, that makes. I'm not gonna lie. Again, I have like virtually no experience with rodents. But, like when I watch the video, it's like, yeah, they just seem like super like frenzied. Of like, I'm sort of like picturing of like, well, what's that movie or that film?
Requiem for a Dream?
Well, there's that one, but like, the person, Willard, is it, with all the rodents?
Oh.
I
don't know that I've seen that, which is weird. I've seen most rodent movies, but I think I know what you're talking about, though.
Well, yeah, like, I thought this is more like a movie thing of like, oh, just rats like super frenzied, and like their behavior, but like, I didn't know how it actually tracked in real life. But I also understand that, they're being manipulated at the level of the brain. But it's like, yeah, super cool. So I think that covers more or less like this first experiment of pretty much the two conditions, basically the effect of the laser on how they consume sucrose, in a nutshell, right?
Yeah, how they consume, I would say in this case, a natural reward, because food, especially sweet food, is inherently rewarding to all organisms that eat in such a fashion, because it's evolutionarily adaptive to get calorie and whatever rich type of food.
Yeah. And I do have a question from chat, even though we touched upon it like earlier, but if you want to like maybe if you're able to unpack it more, or even just say the same thing of like, so basically, I'm sure you're aware more than I am of like maybe limitations of animal models or how animal models are criticized, but like, do you truly believe that this is relevant to humans, or like has the potential to be relevant to humans?
Unequivocally, yes. Yeah,
Okay.
100%, absolutely. I do very much appreciate that, at face value, this does not look like lighting up a cigarette or injecting heroin, right? At face value, the face validity of this model is nearly null in a lot of ways. So I recognize from a lay audience that's something that's very logical to think of as a critique. But when you get down to the level of the circuitry of the brain, it is indeed the best we've got, and it's pretty dang good, especially when you tie this with functional neuroimaging in human individuals. And there's actually a research group at McGill University in Montreal, Quebec, that does imaging research with individuals that are addicted to cocaine, and looks at their response to cocaine cues. So this has been scaled up in a translatable fashion to a human model and has held water. So it's not just so much that it does indeed, I think, work. It's that it's kind of been shown conclusively that it does function in this way. Of course, with lots of nuance. But yeah.
Yeah. So if I'm again correct anything that I say is incorrect. So like, yeah, we haven't done optogenetics in humans yet. I'm sure it's coming in the pipeline. But using, let's say, more orthogonal techniques, we have essentially observed similar results, maybe identical results, at the level of humans.
Yeah, I would say it's certainly not with intracranial opto stimulation. That's certainly not been done in humans, as you said. But doing a data collection using a functional fMRI. they've basically gotten individuals to interact with drug cues. So these would be the equivalent of the lever in this situation. They're interacting with those sorts of things in the paraphernalia associated with the drug. And they're measuring the activity in certain areas that we've been showing in rodent preclinical models are, we think, very relevant to addiction. And so those are the ones that are upregulated in the human studies. So we think that that's a pretty logical leap to be able to make. And until we can actually stimulate these areas ethically in human beings, it's going to be more or less impossible to really get at that. But of course, you can look at things post-mortem. You can look at sections of brains. You can look at lesion studies and stuff like that were done historically, and those sorts of behaviors as well. So there's lots to kind of inform this from different angles, I would say.
Yeah, and they sort of have a follow-up. Well, I'm probably not going to ask it exactly how they word it, but I think what they're hinting at, or what they're trying to tease out, is like basically maybe do we have precedent of like, oh, in this other maybe totally separate area in neuroscience, we've used rat models successfully, and it's been translated to humans, and it's maybe led to maybe some sort of therapy in humans. Do you know if there's any sort of, not to put you on the spot, but like if you're able to say of like basically, do we have examples historically of like, oh, this rat model for maybe this other condition or other thing that we're area of a study, it ended up being applicable to humans, essentially.
So I think there's vast, almost uncountable examples that I could cite here. Probably the most salient of them and the most relatable are those related to pain. So preclinical pain models, of course, are some of the most difficult to do in terms of compassion and ethics. But are the ones that have led to the most huge bounds and strides in pain management in human beings, right? So things like treatment for neuropathic pain, which is related to shingles, is related to diabetes, is related to nerve injuries. These are very, very common things, and pain is the thing that's presented most at doctors' visits by a landslide. And we've discovered molecular targets and pharmacological targets in preclinical models that then go into FDA trials in the United States, or the analogous bodies in different jurisdictions, and get tested in human beings, and are indeed the pain management tools that we use, both pharmacologically and in other regions as well, when they look at, I don't know, like homeopathic things have even been researched in that area, like acupuncture and that sort of thing. So
yeah, that is my understanding as well. Even though, like, I'll be the first, I'm not gonna lie, I tend to be pretty heavily critical of animal models, but I understand like it's sometimes just the best we have to use on offer. But like we also do have, like, as you said, countless examples of like, hey, not that this is a specific application, but we do have good historical precedent of like it's worked for other for other conditions or diseases, or to understand other things about humans. So as of right now, there's not a whole lot of reason to believe that it can't be applicable here. Maybe the overall message.
absolutely, yeah.
So I think we covered that first experiment, and then the second figure. I have to like now. Right. So here, if I remember correctly, I'll probably let you unpack it better. But I think here, they're pretty much, I think, doing the same thing. Or like, I think here they're assessing how frenzied, or their willingness to put in more effort, if I remember correctly.
Right. So if you look across the bottom, the x-axis here, where it says day and effort and FR1 and all these, letters and numbers that are meaningless to anybody outside of the field, what I can say is that these basically indicate the number of actual actions they need to make on the lever, the number of times they need to press the lever in order to actually gain access to that sucrose pellet. So it starts out that one press equals one pellet. And then one of the ways to assess if something is indeed instigating motivation to pursue something, which is, addictive craving behavior in its most maladaptive form, you have to make it more difficult, because People don't have unlimited supplies of cocaine like Scarface. They have to go and seek these things out. And sometimes at great cost, they have to sell things. They're skipping work, they're facing negative consequences, but they also have to go further, farther, etc., to gain access to it. So this is our best attempt at modeling that here, by increasing the amount of effort that's required to obtain the reward. So FR1 is fixed ratio, one response, one pellet. FR4 is fixed ratio 4, four responses, one pellet. Random ratio 4 means anywhere from 1 to 4 will reinforce it. So sometimes it's 3, sometimes it's 2, sometimes it's 3 again, and then 4. It's random, but at a max of 4. And then, of course, RR6 is the same, but with 6. And basically what this is showing is that if you increase the amount of effort that's required to obtain the reward, and if you make the reward unpredictable, that's not going to place anything in the way. So it is indeed motivated behavior.
Oh, one moment. Oh, sorry.
No problem.
Okay. So it's like, yeah, they're basically if I guess to use the example that you gave, I guess maybe of like I guess at the more crude level of like whatever, a drug seeking behavior, although if that's
Mhm.
like not an appropriate term.
Yes, no,
Um
that's nail on the head. Absolutely.
where it's like how far are these rodents willing to go to get that, literal sweet reward of sucrose? And at
Yes.
least based off of what by the blue line of like, yeah, the more they basically illuminate those regions of the brain, the more effort the rodent is willing to put in to get the sucrose. That's sort of like a good summary of what they observed.
I don't think that they modulate the amount of illumination there. They just make it more difficult, and they say, "Are you still willing to do it? Is it that good?
Oh, okay.
Did this brain augmentation? Does this brain augmentation? Is it powerful enough to instigate this behavior more intensely than just the reward itself alone, or stimulation of these other areas like these other lines indicate here. So this is kind of showing, I guess, a compounding effect that we call sensitization, which is basically you make the body, the brain, all of these relevant areas more sensitive to the reward, to the stimulation, to the drug, whatever it may be, which is kind of an inverse version of habituation, or tolerance. Your body becomes more reactive to things.
Right. So essentially, I guess they're kind of like testing out, have we really habituated or conditioned this rodent to really seek out that sucrose? And if we just like let them put in the effort, like see how much effort they're willing to put in.
Yeah, so in a nutshell, this would be showing that the laser stimulation plus the sucrose is above and beyond any other combination of stimulation or reward pairings that they've done here for all of those controls. So there is something special about natural reward, or reward of any kind, and the stimulation of that area.
And one thing that kind of at least stood out to me, well, might have stood out to me based off of ignorance. And if you're able to say, of like, so, you have in the blue, like, laser with the sucrose, or rather, the experimental rodents that have been, modified and getting the illumination. But then in the gray, one of their controls, it seems like, and again, maybe I'm just ignorant, of like, it seems like control is also pretty high.
So you're talking about the gray bar that's just beneath it, control laser plus
Yeah,
sucrose?
exactly. Uh,
Yeah, so I think that they do go on to tackle that and say that it could be the external, just laser stimulus of any kind.
right.
Uh, Visually is like, ooh, party rave,
Yeah,
whatever. They don't say that, of course. But of course, the sucrose itself is indeed rewarding, and you can see that all of the lines that involve sucrose do indeed go up, and that there is something about the combination of any sort of internal stimulation there and the sucrose. But again, that the other one is above and beyond that. So it's not so much to say that there isn't any other areas or any other ways that you can get at this. There's many ways to cause increased addictive and craving behaviors, but specifically the one of interest here seems to be ultra powerful.
Yeah, no, no, like for sure. The blue, like it seems like it's, as you said, like pretty much above and beyond and totally like statistically significant. But it was like curious to me, sort of as you hinted at, is like, oh, are they just responding to that rave-like atmosphere, so to speak? But it seems like they do control for that in like the follow-up experience. So like, oh yeah, like the light was, I guess we'll get to it shortly. Of like they control like, yeah, it doesn't seem like they're responding to the light itself,
Hmm.
and yet despite that, at least for this particular one, which it could just be like an artifact. Like obviously, I don't definitely doesn't like invalidate their findings, but it just kind of like stood out to me like, oh, like it seems like
No, that's a great point.
it seems like they're getting an effect here. Obviously, like it pales in comparison. I was just like super
Yeah.
curious of like
So keep in mind, and I think a really good counter argument to being like, this is a great model of these behaviors. One of the most salient arguments that I hear against rodent models of behavior using operant chambers like that is that's all they have to do in there. It's about a
true.
foot by a foot. And if one of the things makes light and gives you food and there's literally nothing else to do, that's probably what I would do. So that's one of the big critiques of these types of models is they're not exceptionally ecologically relevant in terms of most people aren't put in a room with zero other stimuli and their drug of choice or their craving food of choice, their crux, the pint of ice cream or
Yeah,
whatever.
no, uh,
It's just not how real life works. So that's a really good critique of these types of models.
absolutely. Like I have no problem admitting like heroin in real life doesn't appeal to me, but put me in a room where I only have access to that for a prolonged period of time, and I'm probably going to give into it at some point.
Absolutely, who wouldn't, right? It's interesting. Without sidetracking, a really interesting group of researchers at NIDA in the United States, the National Institute for Drug Abuse. They basically gave a choice model very similar to this, where one lever delivered. They did basically every drug of abuse that's common: meth, heroin, cocaine, you name it. And then the other one gave them interaction with another rat. And unilaterally across the board, what do you think they chose? Most people would say the drug. It's not. They chose the interaction with another rat.
Right. So I think if I remember correctly, and I know this mostly from like Johan Hari, unlike their book, I was a journalist, but like yeah, sort of like early or like addiction research in the past, it was always like yeah, just as you sort of highlighted in this research, like just isolated like rodent or animal, and they only have access to whatever drug that they're studying. But then if they have potentially access to other alternatives that may be equally enjoyable or more enjoyable, sort of like whatever the rat or rodent funhouse. Seems like
Mhm.
that affects how they consume or interact with the drug, or like maybe like they have no preference at all the
Absolutely.
drug.
So Rat Park, I think, is probably what you're talking about.
Yeah,
I hate to sidetrack us even more, but it's a really common thing that lots of people, especially on the Reddit world, really know about. It's a
yeah.
very popular idea. And I believe it was the 70s or 80s that that was conducted in. And it, of course, is a really kind of interesting thought experiment, really cool, and gives a lot of insight into enrichment versus deprivation and how that impacts addiction vulnerability. But keep in mind that most human beings also have a very rich environment, and we still end up getting addicted. So that's a very important factor. But we know because of the reality of day to day life that that can't possibly be the whole story. And interestingly, my master's research was looking at one of the aspects of the story that is as yet uncovered, but that is completely separate. So I will not get into it.
Yeah, fair enough. And I do have another question, if you're able to say, of like, so they're curious about basically the possible influence of concentrated heat added by light, as in, so if I remember correctly, this person's an engineer, and so like they have some insight in terms of like the optical fibers, and the heat generated by light, and sort of their comment is like, basically, I think the power that they're using for the light seems like it might be intense, or like there might be an artifact of heat, as they specifically say, 10 milliwatts in 0.2 millimeters is kind of intense, in their opinion. You know,
Uh,
if like
Right.
it,
So, as far as I'm aware, there is no heating aspect to it. I keep in mind that again, we test these like out in the world with ourselves on light meters and stuff like that. I've touched them right after. I've touched them during. I've tried to like fiddle with them and stuff like that. There's no palpable heat for me. Of course, the brain is much more soft tissue, so that is a good kind of like at face value critique. But keep in mind, if that were an issue, this technique probably wouldn't have got very far. And they've designed that particularly with this in mind. And I'm sure that in the early stages of developing this technique, they probably ran into those issues. And I could probably ask Carl if that's a thing back in the day. But I don't know enough about like light and physics and heat and that sort of thing to really speak to it. But it's not as bright as the laser can go by a long shot. We are using the very low end of the laser. So though it sounds maybe pretty powerful to like you don't want to like you have to wear protective eyewear. It is a powerful laser. Do not get me
okay,
wrong, but we're not turning it up to 11 by any stretch of the imagination. So it's kind of funny. It's an intense and bright light, but it's not like a laser beam like you think of in your head, like Austin Powers and cutting through, adamantium or something
right.
like that.
Well, at the you could probably like safely say it's not like melting the brain or melting surround. Maybe it is, but like, and also, again, correct me if I'm wrong. Like, basically, like the brain doesn't have any like, there's no pain in the brain. Like you can like poke
There is
around in it.
no pain in the brain. That's a really good point. There is no pain in the brain. We would very easily be able to see if there were any sort of like heat-related ablations. That is a way that people used to do research as well. You would just put in like an actual electrical probe and put current through it and burn a very small area. That's an active way that people used to do research, and we very much are not trying to do that here. So yeah,
Right.
not a concern.
So yeah, even obviously you can never say for sure, but they're sort of like speculating that there's probably like some sort of like mild temperature change, 2 or 3 degrees, and they say that might subtly influence conductivity, or like might be affecting, I guess how the ion channels in the brain actually like operate. But I guess if it's consistently like if all of the mice, both the experimental and the control, are getting the same heat, that hopefully should be controlled for. Hopefully.
Yeah, so I mean, keep in mind that they're putting electrodes in brains that have a control virus, so it wouldn't do anything, and they're seeing what just turning the laser on does in an otherwise unaltered brain as well. There's all of the relevant controls to see if that is something, this individual might be correct that there is a palpable change in temperature that might change the local environment, and that is indeed just the nature of if you're fiddling around in the brain, you're gonna make changes when you're in there, whether you like it or not. And it might be chemical changes, it might be temperature changes, it might be pH changes, and all of those things are absolutely very relevant, which is why we use the appropriate controls to make sure that isn't what's driving the behavior. And yeah, that's something it's a very valid critique.
Yeah, that's definitely something I never. I think this is why it's really good to ideally multidisciplinary, and like having people from various backgrounds like look at this type of research, because like I have my certain perspective, and then like the engineer who like probably has way more knowledge about the physics, and like maybe they actually work with optical fibers and light, so like they know how it behaves and maybe how it affects tissues, and then like you with both like at the level of the brain and the psychology and all that stuff. Yeah, I like multi-disciplinary
Yeah.
aspect. So I think, yeah, I think pretty much the main message of this figure is like the rats they appear to be kind of exhibiting sort of that drug-seeking behavior of like they're willing to
Absolutely.
work for it.
Yeah.
And then the next figure. So here, I think this is largely, well, yeah, it's in figure. Like these are the controls, sort of like the quality control afterward. Although maybe we should start with, or like wrap up. No, I think go in order. Figure 3. So yeah, here, if I remember correctly, but definitely correct me if I'm wrong, like they're pretty much like looking, this is part of the quality control of like basically, do they transfect the certain areas of the brain, and basically, are things working as intended,
Yeah.
as desired?
So that the CFOS, which is what they're describing here, or some sort of FOS, I assume CFOS. CFOS is an immediate early gene that is reflective, presumably, of neural activity in that area. So you look at these brains within a half an hour of the stimulation or the activation, and you can stain for CFOS and see if there is activation there. And basically, what this is, in a very small nutshell, showing is that the activation of that area did the laser light shining in that area did indeed activate the area and increase the activity of the neurons in that area, as demonstrated by the co-expression of the virus fluorescent protein with that FOS protein, that indicator of neural activity. So this is just saying the virus worked.
Right. So as in, like, so we got the virus in the appropriate region, and like things were expressing, and then like we actually got neural activity as we intended. That'd be sort of like an
Yeah,
appropriate way.
yeah, absolutely. And keep in mind, without much specificity in terms of cell type and in terms of circuit type, right? Like they're just saying this area, all the cells, or all of the ones that got infected. And keep in mind, there's glia and astrocytes and a whole bunch of other types of cells in there that they're not even talking about. So this is a very kind of simplified, gross overview.
And so you mentioned the CFOS is sort of like, I guess, an indicator of neural activity. Are you able to, so like here, again, I'm ignorant, in terms of like the nitty gritty of the neuroscience and neural activity, where it's like, are you able to say sort of like, what the CFOS is doing, or like, why is it only in the presence of neural activity? Do you get it expressed? Able to say?
So, this is one of the areas that I truly am vastly ignorant in. I think, you know what? I won't even say what I think, because I don't know what I think. All I know is when I asked what the heck is CFOS, they said it's an immediate early gene that's an indicator of neural activity.
Okay,
Or
yeah, I
cellular
totally.
activity, actually, I believe. So I think that it's like a genetic expression that happens after there's some sort of like a metabolic change in the cell. But do not quote me on that, big grain of salt. I truly do not know the background of how that works.
No, I totally understand. And again, like if you're able to say, and totally fine. If you're not, but like, so because like, the way I sort of understand, and that's why I'm sort of like curious about it. But like, presumably, you know, there's neural activity regularly occurring, more or less, maybe not at all times, or maybe yes, actually at all times. And so like, I'm just curious, like, why is it only like in the presence of light? Is this like an indicator of neural activity? Because like I'm assuming other neighboring things that aren't genetically modified, there's still neural activity there, right?
Right, yeah. So that's why they have compared to inactive virus and laser control in this the B portion of it. So they're saying that when they turn on the laser and there's channel rhodopsin present in the cells, it's more than 150% greater activation of FOS in that area,
Okay.
and more than 200 in the other area. So this is very much a relative
Okay,
measure.
yeah. So
And
it's
that's
like,
really right. So FOS would turn on regardless, because you're right, there's always activity going on in the brain, but we're looking at relative activity.
oh yeah, so it's pretty much like way above baseline, as in this.
Yeah,
This is it would not look like this in the absence of the channel rhodopsin.
that's correct. Yeah.
Uh,
This measure is in comparison to without the channel rhodopsin, or, without, yeah.
yeah. Okay,
So,
yeah. That actually does clarify pretty well. One of my main curiosities. I'm still sort of
mhm.
curious. I'm probably gonna like look at some
Yeah,
point.
let me know what you find out about CFOS. I once tried to Google it, and then I was on a plane, and it was pretty early in my master's, and I was like, this is complex. I don't care. I just need to know what it means, not how it works. But now, at the stage in my career that I'm at, I'm like, I need to know how this works. Ooh.
But yeah, since like I just did like a quick Google or whatever, or like no, it's in this because they stain for, well, yeah, here like they stain for it with an antibody, so it's presumably a protein. And like if it's a protein, like, well, that's sort of like my area of like, I want to know what it is, I want to know where it's coming from, what it's doing.
FOS is a protein, I believe.
Yeah, it should be a protein because they stain because it's showing up red here, right?
Yeah.
And so they're staining specifically like for it, and to like get it to illuminate, like probably is a protein that they're using an antibody. I think from I remember correctly from the methodology, they use an antibody, but pretty much an anti-CFOS antibody to like stain
Mhm.
it and to get it to show up
Yeah,
in the microscopy. So it's like, yeah, I'm totally gonna do some research afterward.
cool.
So yeah, pretty much this is a quality control. Is the
Yeah.
system like working? And it appears to be working as expected. We got them. We infected the appropriate region with the channel rhodopsin, and you get that neural activity from the blue light or the excitation.
Yep.
And then this figure, I also have to refresh my brain quickly. Actually, I don't know why my brain is not acting as expected. I can't remember this figure all that well.
I'm rereading the figure caption here to refresh my memory as well. So this is relating it to their behavioral output, and I don't know how they exactly kind of like match those in terms of benchmark, but in the areas where the function maps for insensitive. Hold on.
Yeah, it's okay. I'm reading pretty much the same thing. Trying to.
So, okay, basically, this is showing that like stimulation, it's specific to the central amygdala from a couple of different views, is what all of these are. They're just saying the same things. These are the same dots, just looking at them from different angles.
Okay. Oh
Um,
yeah. The terminology. Yeah. Well, you're. I always get like sagittal and coronal. Like I never use them. These terminologies
Sure,
outside.
yeah. The way that I teach my undergraduate students when I teach intro to neuroscience or psychology is that if you know anything about astrology, Sagittarius is the archer. So if you just picture how it would go through your brain if you were like pulling an arrow, it would go from your nose to the back of your head. That's a sagittal cut, Sagittarius sagittal cut, like an arrow. Coronal, corona means crown. So
Right.
if you picture like, I think of a ray of sunshine because we tend to think of a crown like this, which is very much not the type of cut this is. But back in the day, the Latin root of that is meant to be crown, like a ray of sunshine off of like a Jesus head kind of thing. Um, so I think about it like a ray of sunshine. So if you had like, a typical sunshine over a horizon, that's the type of cut. It would go from top of your head to down to the bottom of your chin, perpendicular to a horizontal axis. And then of course a horizontal view is cutting horizontally through. So at any plane, just going from front to back.
Yeah. That. So the Sagittarius one. I obviously, that's why you're the expert. I never would have thought to use that sort of memory aid, but that totally makes sense.
Yeah.
Sort of like
I
horizontal.
mean, if you're not into astrology, it probably is just as meaningless as it was before. But I find the most useful one for the purposes of these anatomical differences, at least, is the coronal view, because you can kind of picture the left and the right side of the brain and how it's kind of like got the little dent at the top and everything like that, and that the central amygdala is close to the center midline, and that the basolateral is lower and more lateral, right? So it's intuitive to look at the coronal view specifically, for me at least.
And so like it seems like they're pretty much highlighting in this figure of like pretty much these specific regions, as in pretty much zooming in, like it's not just the CEA, but like this portion of the CEA, and then the BLA. It's not just the entire BLA. It's like this portion of the BLA. So I do have that sort of like correct.
Yeah, from what I understand, these are actually where the ends of the optrodes ended up landing, and generally, what you would want to do is, because we know that light doesn't just go exactly where you put the flashlight, or the any of our experiences with light, it doesn't just stop right as it comes out. It
Right.
scatters, right? So we know that even though the brain is relatively opaque, that it is going to scatter a little bit. So they put the optrodes near the top of the structure that you're wanting to, activate, because then presumably the light bleed is going to go to the more of the area that you're interested in, and diffuse out in such a fashion. And I'm sure that there's been appropriate controls done in like a much more basic science sort of fashion in different labs, but I couldn't say for sure.
And I understand, like, obviously this is like whatever, a figure or a diagram. That's
Mhm.
whatever, CGI. But if you're able to say or like comment, like, is it sort of as depicted here of like, this clearly defined like border, and like, they're sort of like represented as like two separate like entities, of like, this is distinctly the CEA and this is like the BLA, and like
Mhm.
there's no mixing, or like interconnectivity between the two. At least this is what I would be gathering from the figure. Is that actually like true?
They're functionally distinct for sure, but functionality, as you're alluding to, does not necessarily lead to visual distinction. They are a little bit visually distinct when you look at, like, say, a Golgi stained brain, which would, stain the Golgi apparatus of the
Yeah.
cells, blah, blah, blah. So it comes out purple, I want to say, or is that, I can't remember. It doesn't matter. Crescent violet, any of these stains that would just stain for cell bodies, or other kind of clear things. What you see is actually this extension from the top here, that kind of arm that's extending down to the box there. That one that. Is coming out to the right of this gray structure. That's an extension of what is the hippocampus, and at the kind of more ventral bottom end, so closer to the lower part of the brain there, where the BLA bottoms out. It actually kind of branches out like this, and it's
Okay.
not patently obvious here, and it depends what slice you're at in the brain, but it actually kind of nicely nestles the BLA, and then the CEA is on the other side of that, and it's just a really thin little strip, but it's kind of like a curved little like pincer sort of looking thing, depending again where you are in the slice. But for the most part, these structures don't go super long longitudinally, sagittally. They're kind of a small little almond shaped nucleus, and together they are, that is.
Yeah, that makes sense. And if you're able to say, because like, so here is where it's like, my rudimentary understanding of the amygdala, even though like I know generally speaking, like, these are just like general brain regions, but like, does basically, does the CEA and the BLA, does that represent the entire amygdala, or are there other like substructures as well, that like are part of the amygdala, but obviously they're not interested in it here?
Yeah, there are many ways to slice the pie that is the amygdala and any part of the brain, and it really depends on what you're looking at. So there's functional separation, there's anatomical separation, there's cell type
Right.
specificity separation. It really depends on what you want to draw the line. Like when we talk about like gross brain anatomy, we talk about gray matter and white matter, and then if you want to parse it by different things like motor and sensory, then there's within the white matter differences there. So it really depends on how you want to slice it. For the purposes of most people's understanding, this is most of the amygdala. This is all you really have to worry about. Amygdala researchers, say at like MIT and Scripps and stuff like that, they tend to parcellate within these areas. So there's the medial aspect of the central aspect of the amygdala.
Yeah, I'm sure you
And
can like just bisect, and probably maybe at some point we'll get back down to like cellular level, like this cell here in the amygdala, or like this collection,
yeah,
and be like, well, these collection of cells in the amygdala, that however we name it, is responsible for this. I guess
yeah,
that's the ideal that we
that
want to
has
move
been
towards.
done, and it's almost out of my realm of understanding, even being a neuroscientist who's relatively well versed in this, because it's really nuanced and sophisticated research that's super valuable, but is very intimidating to say the least. So yeah, you're right.
But yeah, from what I like, this is just my understanding of like, yeah, they want to, I guess, that perfect understanding is like at the like most high resolution cellular level. It's like, yeah, these cells, like, they do this, and we know exactly what it is that they're responsible for, and then like just going like almost like cell by cell, or like collection of cells by collection.
Yeah, and you can do that. You can do that, and you can even go to the receptor level and do electrophysiological recordings in that area and
True.
get
Yeah.
You can get super nitty gritty, and they use opto. Actually, that's a good answer to the temperature one and how it would affect ion flow at the level of the specific channel. They use opto on in patch clamp electrophysiology, where they're looking specifically at one of these ion pores or receptors, and it works just fine and is analogous to any other thing. So I think that that's a pretty conclusive thing to say that the temperature isn't doing anything super important for the purposes of our understanding.
Yeah, like obviously you can never say for sure, but like I would have hoped,
Yeah.
again, this is like a really popular technique that er- everyone wants to use more and more. That like if something is, you know, I'd be not anything's possible, but I'd be very surprised if something is if someone had to consider temperatures like, oh, this like destroys the foundation of optogenetics research. But
I
anything,
am certain that someone got in there and turned it up to 11 and was like, "That's too high," when they were developing it. I'm confident that that happened, and I'll actually ask and see if I can get back to you with an anecdote. But yeah.
but I actually, and well, I guess again, with getting too sidetracked, I almost feel sorry for like the early rodents subjected to this. Like, oh, I understand it's science. Like, oh, we melted their brain. Let's
Well, they
dial
would have
back.
done this in culture and in cells before they put it in mice. Like people do their due diligence, and you're not allowed to just willy nilly do that sort of stuff. There's strict regulations, even in the pain research world, about the type of pain that you can put them in. We can poke them with a little thing until they lift their paw away, but we can't like chop off their hand or you can't do phantom limb models in rodents.
Right?
You're
Yeah,
not
that makes
allowed,
sense.
right? You just can't. And that's a very relevant thing that it would be nice to be able to research in preclinical models, but it's just simply not ethical and no one would allow for it, rightfully so. Right?
Yeah, that is reassuring. I'm not gonna lie. I wouldn't be able to do. There, I went into molecular biology because like I can't work with animal models. I don't really have the stomach for it. But I recognize its utility and I totally respect it.
Yeah.
But it does kind of
No,
like some.
it's not easy, and I think it shouldn't be easy. And I think anybody that does it, that does animal research and doesn't struggle with those things, shouldn't be in that field. I want people like myself that when it's the day that I have to extract brains, I cry every time. And I think I should. I think that that's I if I didn't, what kind of heartless monster would I be, you know? So yeah, as much as I wish that I didn't have to do those things and that that weren't, a logical part of it, I would much rather have me who has worked at animal shelters doing that work and making damn sure that that animal is not suffering any second longer than it has to.
Yeah, that is reassuring. And here's sort of like so. Um, yeah, one of the reasons I figured out pretty early on, even though I knew like way before I went to graduate studies that yeah I won't be able to work with animals. Um, it's like so I got like a little bit of a hint of just how, at least for me, kind of like how creepy it gets, at least based off of what I observed at my institution. Of like I had friends or rather have friends that are neuroscientists, and like they do research on rodents, and like they gave me a tour of like the facilities. And at least at this institution, like the facilities were like underground, like hidden away, which as it should be
Mhm.
like for security. But then like like it was just like when showing me how the rodents were caged or stored, it was like it's like this like white corridor and like with a bunch of doors, and like behind each door it was like filled with just like cages of rodents. um I think mice at the time. And it just seemed like sort of like something out of a movie of like just like this white corridor
Yeah,
hallway, and then behind each door uh, like literally like I
yeah.
don't know, hundreds of cages of mice.
Yeah, yeah, and that's more or less the case, because you want to humidity control it, temperature control it, make sure that there's not pathogens getting in. If they have sentinel animals and that sort of thing. It's very rigorously controlled for temperature, for the type of food that they're getting, their air filtration. They arguably get it more controlled than most people that are exposed to asbestos in the buildings and stuff like that. In a lot of ways, but I do recognize that it's not savory to experience. I completely acknowledge that. Yeah.
uh
Anyways,
that's okay. And again, well, I'm the one that sort of sidetracked things. um
no.
So, yeah, this other experiment, I think it's roughly the same or like similar to the previous, where Well, here I think it's like a single session. I think the other one was like over a course of like days, and
That's right.
here it's like on the x-axis, just like one 30-minute session. You know, how hard are you willing to go? How seeking? Oh yeah, pretty much. How much effort are you willing
What's the
to?
intensity of the drug seeking? Yeah, that's exactly correct. So your instincts were directly on point. You're way better at this than you're giving yourself credit for. Like you're everything you're saying, you're like, here's a simple way to say it. And I'm like, yeah, I could have said it that way. Hey,
Well, I appreciate. I appreciate. I don't take that for granted.
yeah, well, I'm sure it didn't come easily, right? You know, it's not effortless for any of us to understand this stuff. But basically, what you have, as you said, on the y-axis, of course, is time, and this is a single, what we would call a challenge session or a probe session, or something like that. And before I was talking about a fixed ratio scale where they get one reward for one lever press or one reward for four lever presses. In this case, it's what's called a progressive ratio scale of reinforcement. So mathematically, it progresses. There's different models of progressive ratio reinforcement, but most of them go up in an exponential fashion. And basically, it just becomes increasingly more effortful to get the same amount of reward in most cases in a progressive ratio schedule. So you can see on the right-hand side, the price is the number of times they have to press the lever to get one pellet, I believe.
Yeah.
And you can see that it adds up very quickly because it starts off as one press and then usually goes to two and then doubles from 2 to 4 and then doubles from 4 to 8, etc. I think they actually have a bit of an adjusted model based on an exponent, and I am not good at math, but the point is it gets harder and it gets harder fast, and it's a steep curve. And this is basically saying that they're still willing to put in a lot more work to get a minuscule, unchanging reward.
Yeah, because as highlighted, well, pretty much on the y-axis is like up to, well, at the maximum like 800, and this is like cumulative. Like they're really pressing. But so, and also you can break this down as like a rate because you have the time. Like this is like at over 30 minutes, like to get 800
It's
presses.
nonstop.
Like yeah,
Non-
uh,
Only a stop to go and get the pellet, probably. But
yeah,
having watched this and done this in rats that were self-administering cocaine and remifentanil, which is a really short-acting opioid, they'll do exactly that. They'll do it, and they'll in lots of cases you have to build in a timeout so that they don't overdose themselves.
yeah. And I'm just like picturing with that frenzy behavior of just like they're just like it's almost like a workout. Like I don't know if rodents sweat, but I can imagine like
Funnily enough, one of my coworkers researches how exercise changes inflammatory response, how it relates to pain, and they get mice to climb up this little ladder, and the mice absolutely love doing it, and that's their little exercise that they do, and they get muscular gains. We like measure
yeah.
them and stuff like that. It's really cool.
But like yeah, they're going at like whatever, like 30 presses per minute at some point, or like yeah, 10 to 20 presses per minute. So like
It would be
that's
nonstop. Yeah,
uh
non.
and not to mention like they got to consume the soup. Well, presumably they want to consume it, and I guess they're consuming it like pretty quickly. They're like
Yeah, it's a very small pellet. Like it's I don't know. I'm trying to think of something that would be that size, like a BB pellet, maybe.
yeah, because I do remember from the paper, it's pretty much like in the milligram range, and I weighed out
Yeah,
milligram, like it
8
is,
or 25. It's one of the two, and I know that no, it's 25 for rats.
yeah,
25 mil.
and I
It's
know
tiny.
like yeah, it is, that would be tiny, but I think I wonder, I'm curious, um, do they control to that level of like calorie consumption per sucrose pellet, and like how that's maybe a like confounding factor, like these rats consumed whatever 800 calories of sucrose versus the controls
Yeah,
uh consumed
that's a
less.
really. It's funny that you're getting at things that a reviewer would get at here, and that's a really important consideration in a lot of these things because, as I was talking about earlier with sensitization, making the system more sensitive to the substance, the drug, the food, etc. More of it begets more sensitization. So it starts to become kind of a self-confounding thing that the ones that took more, maybe just because randomly they took more at the beginning or pressed the lever faster or whatever, or they, hadn't eaten as recently or something like that, so they weren't as hungry, maybe just by happenstance, those ones just took more and that changed the brain, so that made them want it more. And that is relevant because that is how things work in real life, but how do we know that that's not what's driving the behavior? And the way that a lot of people tackle that when they think it's a, a potential confound, is they do what's called a yoked design, where they just basically
Okay.
give the animal, in this case it would be drug, comparable to what the other animals would have gotten, so that they're getting the same substance but the action isn't the same, so that you can parse apart any differences that would be there, would be specific not to the amount of the drug, but with the cues being paired with the drug, which is again that really important component of the incentive sensitization theory of addiction specifically, that those cues instigate that craving state.
Yeah, that makes total sense, and it's good that I guess they take. It is good that they take that into account. But I'm also like again not to get like sidetracked, but like
Mhm.
I can sort of imagine that you consume enough sucrose, there are metabolic changes that occur.
Yeah.
That we
It's
know this
called
definitely at the human level.
cross-validation. Yeah, there's a word for that, and it's a very interesting area of research. Cross sensitization across polypharmacy is a real thing. When you think about addicted individuals, people that are addicted to heroin are usually smokers and
Yeah,
drinkers. They're not usually hitting the gym and then going home and doing their heroin like American Psycho style. Or I know he doesn't do that in the movie, but you know what I mean? It's not usually parcellated into that part of life. There's lots of bleed through into other things. And we think that the reason for that is exactly what you're describing, that there are changes metabolically to your body that may relate to the changes in the brain or may not, likely would. But there's changes in those areas of the brain that are related to rewards, because rewards come in all sorts of shapes and sizes, right? So sensitization from food reward may translate into the same type of sensitization for other rewards. So you hit the nail absolutely on the head, and they do not address that. It's borderline impossible to address
yeah.
without making that the purpose of the study. And people do study that. Not this group, though.
Yeah, no, I know at the end of the day, like especially with complicated research like this, like it's impossible to control for everything. As opposed to like for me and for some of my experiments, not all of them, it's like I could control like virtually every like parameter and variable. Because like these are usually like things I'm not working with animals, I'm working with like bacteria and proteins, and so like I have just like
Mhm.
much greater control
Yeah.
over the variables. So it's like, yeah, I know that there's always like an asterisk sometimes with this research, but like if you
Mhm.
want to control for everything, it's a Herculean task, which is like impossible.
Yeah, and it starts to self confound itself, and then it gets even the more you control for things, the further it gets from reality, too, right? So we really want to balance some sort of ecological validity with realistic control of confounds or potential confounds, right?
Yeah, exactly. So I think the main message, at least for that experiment, is like these, the rodents or the rats, like they're willing to work really hard, or at least the ones that has essentially been conditioned, like with the laser, they're able to like work
Mhm.
really hard and exponentially hard because, like as you said, like it gets more and more. Like the first one is 2, then it's 4. It's like a doubling where you're just
Yeah.
getting like lots and lots of presses for just like the smallest of reward, or like I guess not to be too crude, but like
Absolutely.
the smallest of hit, so to speak.
Yeah. Yeah. That's a great way to put it.
And then for this one, so this one was one of the ones that was like sort of like most interesting, or at least maybe the most informative to me. But again, I'm not in the field. But like that, when I was first starting reading it, I was like, oh, I wonder how this would compare to like this of like not an actual reward, but can they turn something that's not a reward into a reward? That's essentially at least my
Usually,
understanding.
a neuroscientist, your instincts are very on point.
I appreciate that. Where it's like, well, it seems like that's what they're testing here. Like, well, they presumably shouldn't like salt, and so can we basically turn salt into sugar, or rather have them behave like it is the sucrose? My understanding.
Yeah, that's exactly it. And it's funny, this actually paves the way for a really elegant paper that Shelley put out 2 years ago, who's the second author on this paper from the same lab before she moved to do her postdoc. And it actually showed that you can transform a painful shock probe. So, like a probe that they voluntarily walk up to the first time and learn that, ooh, that gives me a little shock. I don't like it. It's the equivalent of like a static electricity shock. So, mildly aversive and but not super painful. Um, and they can condition, just like they do with the salt here, approach behavior towards that with channel rhodopsin excitation of the central amygdala. So this is really paving some ground for some further, even more profound and influential work that this group does. So yeah.
And what stood out to me, or like what's, even though like it's, they get really significant findings in terms of like the effect of, let's say, the light and the conditioning. What sort of like stood out to me is sort of like the outlier of like, it seems like every once in a while, like they would prefer salts, even in the app, like as in like not like a binary of like
Mhm.
some rodents, like they willingly go to salts even like in the presence of like the laser, right?
Yes, yeah. So I think what you're getting at there is a really another great kind of subtle point that kind of circles back to those earlier theories of addiction that we were discussing, and the fact that there is some role for the actual hedonic liking value, because liking and wanting are very dissociable things that typically go together but don't always go together, and that's a really important point of this group's kind of assertion and theory of addiction, is that they're dissociable. But what this is suggesting is that, yeah, you can turn something that isn't normally rewarding or enjoyable into something that is rewarding and will be craved and pursued. But what you're noticing is that it's not as powerful of an effect as it is when it's paired with something that is in and of itself enjoyable and intrinsically craveable or wanted kind of thing, as well as liked. So that kind of hints at the fact that we don't tend to get addicted to things that are at their outset immediately already aversive. There is something important about that hedonic thing initially that does tend to fade out.
And I'm curious, if you're able to say, because like that is, as I said, like stood out to me of like, so, you know, for myself, put the two choices in front of me, like no matter what, put salt, put sucrose and salt in front of me, I'm choosing sucrose 100% of the time.
Yeah.
And so it's like, I don't know the natural or like the unmodified behavior of rodents, but is it like maybe like rabbits, like they sometimes like want salt? Um,
So only if they were mineral depleted, and they've done that exact study where they deplete sodium out of the body of the rat, and they will pursue without any other interventions. They will
yeah,
go
that
and
makes total sense. Because, well, yeah, they need to survive, right? At that point, if they're
yeah,
starved for salt, like they want to live,
yeah.
or like there's that strong craving driven by survival to like consume salt. But here it's like, it seems like they're voluntarily consuming salt, as opposed to like, yeah, put those choices in front of me, like it will be a binary pretty much.
Yeah,
uh
I mean, keep in mind that if they have just recently drank a whole lot of water, that there's probably some osmotic balance that would be really finely
Fair
a
enough.
lot. There's a lot less margin for error when your body is smaller, right? So when you're 30 grams, or in this case, they're probably 500 grams. For I think they have Sprague-Dawley or Wistar rats. They're typically females and males kind of are anywhere from 500 kilograms and 200 sorry, not milligrams, 200 grams plus or minus that. Your margin for error, if you just, chugged a bunch of water, would probably, throw your electrolyte balance and your osmotic balance and pH and all that kind of stuff out of whack pretty quickly. So it could just very much be a proxy of individual differences in their state when they go in.
Yeah, so yeah, this is why like I love consulting with actual experts. That's something I wouldn't have actually considered. That totally makes sense. That totally tracks.
Yeah.
Of like, hey, if you're just like consuming a bunch of sugar and you're probably, I'm assuming they have access to water, where it's like, yeah, osmotic
Oh, yeah.
balance. That makes total sense, because like, yeah, when I was reading this, and like as they highlight here, 3% salt, that's essentially like ocean water. And it's like, that's a very high, like, I can't imagine ever wanting to like consume that, unless like, yeah, even like in the most really hard to imagine, but it makes sense of like that osmotic balance if they want to maintain it. That's why it's that stood out to me of like, oh, they're like voluntarily consuming this really salty stuff.
Yeah, and I mean, I'm just postulating. It might be something completely unrelated, but I would be shocked if that weren't a
Yeah,
likely
you're
explanation.
almost certainly right, which is why like I want to ask, have way more insight
Mhm.
into this. But yeah, it seems like for they basically are able to turn something that, well, generally speaking, shouldn't be a reward into a reward, as in it looks like they're pretty much preferring the salt pretty heavily, at least in
Yeah,
comparison to like previous.
yeah, yeah,
Um,
exactly.
and I think that's pretty much like the main message. Uh,
Yeah.
yeah.
So yeah, I guess if you zoom out, I mean, the take home message for that is that the central amygdala is driving craving. You know, it's possible to do that, and it's not just that the sugar needs, or that the CEA needs the sugar, though that is more powerful. It's that they're saying like quintessentially we can, you know, it's necessary and sufficient to create this phenomena.
And I don't want to. I was. I'm gonna like go on my own deep dive of like the like left to their own devices, how much salt just uh
Yeah, that's a
do.
good point. I'm sure veterinary scientists have done that. There's lots of research in these areas. So.
And so here for this figure, one of the last ones, if I remember correctly, like pretty much this is like their one of their controls, or like quality
Mhm.
controls of like trying
Yeah.
to control for like that sort of artifact of like light exposure, as in like
Yeah.
they're generating sort of like rave like conditions, and maybe
Yeah.
it might be like just hyper stimulating them, and just as
Mhm.
in a rave, like not that I have much experience, you want to have a good
Yeah.
time, why not have some sucrose? Uh,
Yeah.
and so here, if I might remember, well, I guess in the figure the way they do it, correct me if I'm wrong, where it's like, yeah, they have pretty much that square, and then the center is blocked out. They can't go into the center, and they just track how they go into the corners. And like in one particular corner, as highlighted in the figure, like they get the pulse of light or multiple pulses of light, and
Mhm.
presumably like if they're responding to light, they would spend more time in the corner, but since they observe no corner preference, it seems like the light does not appear to be doing anything. Sound right?
Yeah, that's exactly right. So it's one saying that it's not the light itself, the visible light from the that's able to be observed by the animal that's driving the effect. Um, and this is what we just would call a place preference test. The funny thing is that if it's driving reward, it should create place preference, because that's another canonical way that we measure if something is rewarding or not, or at the very least unaversive. But I think the other kind of take home point that's maybe less obvious here is that, again, the presence of that external reward is necessary for the central amygdala to be relevant to the reward process. If there's no specific, discrete, observable, localizable reward present,
Right.
then there's nothing to assign valence to. And remember, at the very beginning, I was talking about the central amygdala being associated with assigning the goodness or badness of things, or encoding for that in its polysynaptic connections. And you can imagine that, yeah, it's probably really good at that. And when you crank it up to 11, you can turn something gross like salt water into something rewarding. But if there's nothing to turn up to 11, or nothing to focus that energy on, then it doesn't in and of itself create, an intrinsically rewarding state for the animal.
Yeah. That's something that basically I hadn't like fully considered. Where it's like, so it seems like that the intrinsic rewarding aspect of the sucrose is like pretty much like that necessary condition for to drive the behavior. I guess that whatever drug seeking behavior, as opposed to like, hey, if it's just hanging around, it's like, well, I guess there's nothing intrinsically rewarding of just like being in this spot in this corner, so they're not
Mhm.
conditioned to it, or they're not going to be uh
Yeah.
they won't be displaying corner seeking behavior basically.
Yes, yeah, exactly. And I would just like remind people that also it works for the salt too. So it's just that it needs an external focus
Right.
location, or something to direct that energy towards. It doesn't need to be something that they like. It doesn't need to be enjoyable for it to be a target for an addictive, like craving, a drug seeking, or sensation seeking behavior.
I'm curious. So yeah, if you say they need something to focus, I'm curious because like if I remember correctly, it's not obviously diagram in the figure, but like
Mhm.
the rats are on plexiglass. It's like a clear floor, if I remember correctly. that they're wandering
You
around?
know, I didn't breeze through that recently enough to say for sure yes or no, but I can take a look. You're talking about this for this place preference one, hey? Procedure apparatus. Instrumental, stimulation test, reactivity. Okay, so that's the taste reactivity was in a square plexiglass chamber, but I don't know about the place preference.
Not that it matters much, but I wonder, like, because if you say they need something to focus, could you just put something as simple as like, I don't know what the equivalent of like a rodent toy is, or even just like something like really small and trivial,
Yep.
of like a Lego block. Would that potentially change? Or like, oh, they now have a preference for this because there's something to focus on, if that's what it is. Uh,
I would say that almost certainly they would. There's something kind of related to that called the novel object test, where you can put them in there with like two, pyramid shaped blocks, and then you switch one of the pyramid shaped blocks out for a square block, and you see how much they interact with the novel object, and that's supposed to be a proxy for exploratory behavior. That's sometimes related to this type of thing. And yeah, right, rats are, they have both a neophilic, neo meaning new,
yeah.
philic meaning philia. They have a neophilic drive, but they also have a neophobic drive because they're prey, right? So they both don't want to explore too much because they don't want to die, but they don't want to not explore at all because exploration is how you discover food and mates. And new territory for habitats and stuff like that, right? So they have these competing drives, and in absence of other stimuli that are competing, they probably will go for a new thing. And if you put new thing in the corner with this stimulation, I would hypothesize that they would then be like, sweet, Lego block, love that, super rewarding.
Yeah, I would have been like again. I know that at some point you have to put a limit on the number of experiments that
Yeah.
you run, and I know that there's always that balancing act of like acquiescing to the reviewers.
Hmm.
But like I think that would have been at least for me like sort of like a nail in the coffin of like, oh yeah, we just like put something to focus on.
Yeah.
Then it's like, yeah, they'll prefer. You can turn a no preference just as they did for the sucrose, turning it to
Yeah.
salt. You can condition a preference for and sort
Yeah.
of like see the same effect.
Well,
Uh,
that circles a little bit back to animal ethics if you think about it, right? Because, yeah, it would be really nice to see that you could kind of show this in a second way. And sometimes we do have to do that to kind of really assure ourselves that the phenomena we're observing is explained in the way that we're explaining it. But at some point, it crosses the line into beating a dead horse and wasting animal lives and time, right? So we really have to be careful about balancing that, both in terms of, actual time and effort resources and the bounds of reality, but also just like what's appropriate to make a mouse do and, use mice to
True.
do, right?
Um, yeah, absolutely. And um, as you sort of like highlighted earlier, as we went through like the methodology, like they transfect the region of the brain, but then if it goes on long enough, like the entire brain, if I remember correctly, will essentially like be transfected, and so
Mhm.
like that, basically it seems like you have a short window to run all your experiments once you started on the path of like modifying these mice, right? So you got to make sure, pretty much, I have no doubt that have to assume that everything is well planned out. Like once we start, this is what's going to transpire, and then like that's the plan, and so we can't really deviate from that. That sort of.
Absolutely. In addiction research, it's a labor of love, and it's not exceptionally researched in these behavioral ways for a reason. Because you have to do it. Rats don't care if it's Christmas or Hanukkah or Eid or anything like that. They don't know. They do not care. So if you're designing an experiment that's 21 days long and it goes over Ramadan or your birthday or what have you, it doesn't matter. And you can take a break, sure. The rat's not going to really care unless you put it into withdrawal from whatever drug it is. And it usually they enjoy these sorts of things, so you might make them bored, but that would be a big confound for your experiment. And there's other phenomena that are like withdrawal related cravings and intermittent access that are played out as well and are a big part of the research that people do in addiction world as well. But yeah, you would you need to do it day after day after day. For my master's research, I slept in the lab for days on end.
Oh yeah.
And I had a 45-day long paradigm that I would do. And it was, you're there every day for 45 days.
Yeah, just another reason why like I didn't want to work with animals. Like I work with bacteria. Like if my batch of bacteria happens to go bad, not like they're usually pretty robust. It's like, well, I can just generate a new one in the next couple of days. Their
Yeah.
generation time is like 20 minutes.
Nice.
And
Yeah.
like yeah, whatever. If I my purified protein, I could just like put it back in the freezer, and generally it's like good to go day
Yeah.
or like next week.
Yeah,
Uh,
not the case here. I that sounds very nice, but it also sounds like something you need to be really precise about. Not that you don't need to be precise about the brain surgery part of this, but I like to be able to pick the thing up and like kind of scratch them behind his ears. I don't think you can do the stick your finger in the petri dish and scratch them
No,
around.
so like that's
Might be
like
a
I
problem.
would totally appreciate like interacting with a rodent and like subjecting them to these tests like seem really cool, and I would like that. It's all the other stuff of like the
Mhm.
dissecting, the sacrificing, that if I can like offload that, even then I would probably be like, no, don't do it, and I would invalidate the research. Um,
Yeah.
but like yeah, working with proteins, like yeah, like I'm just staring at like a clear tube or clear solution. There's other challenges working with proteins in terms of like stability
Yeah.
and like so actually there's maybe just as you need to keep the rats in a particular state and happy and controlled conditions, that is actually applicable to proteins. But like once you found like the appropriate storage conditions that made them happy, like you can just like keep it in the freezer and it's usually good for quite
Mhm.
a while. Uh, but I don't want to. Not that it's a competition, but there are totally challenges when it comes to working the protein. That totally can be a headache. Definitely not like
It
walk in the
sounds
park.
like even just working with the virus that we use in these situations, they denature when you
Yeah,
defrost them out of the
exactly.
-80. So there's a whole bunch of considerations there. And even that tiny consideration, I'm like, this is a nightmare. I could never do this. So I very much admire that, there's people like you that do that because that is not my bag. Pipetting and stuff like that, that other people do too.
Oh,
I'm like, oh, my thumb.
I'm not gonna lie, like pipetting is like one of my favorite. There's just something therapeutic of like
Really?
transferring like a specific volume of liquid from one tube to another. Like it might just be an OCD or something, not to misuse the term, but just
No,
uh
you're obviously in the right profession because I hate pipetting so much.
no, like it's like you just if you're like listening to something, like I can like get in a zone, and especially if I'm tracking like hundreds or dozens of samples, it's just like yeah,
Yeah,
pipette go, eject, like yeah, I can get into a nice
cool.
sort of a state of flow, I guess.
Yeah, yeah,
Uh,
no, I get that way when I slice brains, which is much more morbid. Ugh.
no, I mean to each their own. Um, But so I think pretty much like yeah, the summary of like this figure, I think they're pretty much like testing the same thing in both conditions. Like one is like corner preference, and one is basically like spout preference. Pretty much like trying to control out that like the light has no like appreciable effect. They're not responding behaviorally to the light. I guess would be
Yep,
the main message.
yep.
I might have a question for a second.
No
Uh,
problem, no problem. I can't
uh,
see your, so I don't know what they.
Uh, so they're saying just reading paper confused about the position in box preference experiment. They don't seem to say how the rats can differentiate areas of the box. If box is symmetrical, there's no external reference to know where one is. Um, Although you probably I'll just give like my sort of speculation of like I don't think they care about the preference. They pretty much like want them to wander around with no preference whatsoever. But correct me if I'm wrong.
Yeah, the idea is that they're not dissociable for any other reason because like if you put polka dots on the wall in one part and stripes in the other part, which we do, and it's called novelty preference. That's a completely different test, and yeah, you can test for that preference too. But that's just simply not the variable of interest here. We wanted them to otherwise be indistinguishable so that we could indeed say that it isn't the fact that the laser happens in that corner and that's the only distinguishing factor that's causing them to spend X amount of time there. They're just spending approximately equal random amounts of time in each spot, which is what you would expect if the laser wasn't creating its own kind of special aversion or appetitive effect.
Yeah, that was my understand. And well, yeah, not to I guess dissect it too much, but so uh in panel A or on the left side, it's like, so, the main message that they conclude is like the light is having no appreciable effect or
Hmm.
response. But then if you look at like the top one, it looks like they're you can maybe, make an argument that they're rejecting that corner, the 15.6%.
Yeah, I
I
mean, I guess, yeah, it looks definitely visually like there is some sort of appreciable difference. The statistical significance in the difference is certainly not there, or they would have had the
Yeah,
answer
that
correct.
I totally take
But
that
yeah,
point.
it may be one of those things. Remember, their sensory inputs are very much more sensitive than ours in some ways. So I'm sure that the laser actually turning on probably creates a little humming noise or something like that. Like, it could be some variable that's associated with the laser that's like just enough to make them like stop and want to, like, move to a different section and kind of like explore and like look to a different area, but isn't enough to cause a significant difference, right? Because again, we can't control for every
Yeah,
single thing. You could, turn a different laser on at a different time, but that becomes a logistic nightmare. Um, but yeah, I that's kind of my guess as to what would happen here. You can also see that they seem to spend more time in the top left and the bottom right
yeah,
than either of the other corners. Right? So, what's about those? Maybe the lighting. Usually, you control for that when you're doing these sorts of experiments. You have special light boxes. You measure it with a light meter. But the door has to be on one side. Maybe that's where the door is, and that's where the humans come in and out of, and that's where their cage is, so they can probably smell. You know what I mean? It could be any of these
yeah.
things, and
That's a
you
good
have to
point.
have a door into the room. It's not The Sims. You can't delete the door.
Not yet. I'm sure the simulations are already there. Like I know, like
Yeah,
we're working up to that. Um,
absolutely.
yeah, not like I didn't want to be nitpicky, but I
No,
definitely have that
please
side
do.
to me.
These are really important questions to ask, and I think it's good to nitpick. That is the nature of the review process and the critical inquiry and that sort of thing. So yeah.
And then here, I think pretty much like their overall like last, I guess meaningful experiment for the results. It's like basically controlling or attempting to control for that sort of like, as they say, hedonic liking, as in
Mhm.
it's they're not finding the food like more rewarding, or I guess maybe like more tasty. uh,
Mhm.
that it's really like something else as the mechanism. It's not like they're not finding the sugar like, oh, this sugar just is taken to the next level sugar, but like some other thing that's going on behaviorally. If that's correct.
Yeah. So let me just like really clarify that I know what I mean.
Yeah, as I should probably highlight it of like the wanting versus the liking. You sort of like
Okay, so I just wanted to make absolutely sure that this wasn't concurrent stimulation while they were getting these things. This is just saying like when you give them the chance to eat a bunch of M&Ms outside of experimental time, outside of the box, and their actual amount of chow, they probably just weigh it, like their free ad libitum amount of chow that's in their cages. Ad libitum, sorry, that's a super jargony. That means as much as you want.
yeah,
They have more food than they could possibly eat. For those that aren't familiar with the Latin term that we use, blah. Anyways, it's basically saying that it's only applicable when they're doing the behavior. So it means that I'm trying to think of a good analogy, right? That your nicotine cigarette, if you got like, it means that like your nicotine patch, people don't jones for nicotine patches the same way that they jones for cigarettes, right? So it
right.
means that you could give them sucrose in a completely different form, in a completely different context, and because it's not paired with those cues, this is really trying to reinforce the fact that the incentive motivational value of the drug is one thing, the substance, the sugar in this case, is one thing, but it's the cues and the environment around it that really are the thing that's driving the addictive behavior and that motivated craving based behavior. If you were just, in a completely different context, and people can usually report this, when you're at home during the holidays and you're in the holly jolly whatever, if you, follow Christian North American holiday things, then you probably are, around the sights and sounds that make you munch a little bit more, right? So these are the kind of ways that we're trying to model those things. And then on the right, we talked about this a little bit in our preamble. The lip licking is a very interesting way to measure hedonic response, right? Um, I don't
Yeah,
know if you.
so yeah, I definitely wanted to unpack that more, where it's like if I remember like sort of how they basically test for it, it's like pretty much through human observation,
Yep.
through like looking at their behavior, like how much they're like whatever licking their lips, or like whatever pawing or maybe drooling, and they measure the amount of and it seems like yeah, it's like pretty much like through human rated observer, like they score it, like oh yeah, it looks like they're enjoying it, thumbs up,
Yep.
which like it sounds cool. And like I'm totally ignorant on these things. I have no doubt that this is just like a known thing that has been used that probably goes back decades, that
Yeah.
has been like verified. But I do kind of like find it like a bit strange, in terms of just like scoring like rat liking just by like looking at licking that type of behavior.
It's a funny thing, and it's found its way onto meme pages. Their original paper showing hedonic facial orofacial responses because they did it in chimpanzees, and they show that they stick their tongue out and lick their lips. They did it in babies, same thing, and then they did it in rodents to show that it's evolutionarily conserved behavior. And it is kind of funny, and it's kind of like it seems like a willy nilly unscientific way to measure it. But if we kind of zoom back out and think about a couple of things that are instinctive reflexes that human babies before they're trained or conditioned or anything they have several. So there's the plantar or the palmar reflex. So if you touch the center of a infant's hand, it will grasp. If you touch its foot, same thing. That will happen in chimpanzee babies, etc. Right? If you stroke the cheek of an infant, it will open its mouth and turn towards it because it's looking for a nipple that has breast milk in it. Right? And if you think back to, this is a cool anecdote, the cover of Nirvana's Nevermind, which is a baby swimming in a pool, and
Right.
people are like, how in the heck? It's because a baby has an immediate, reflexive, unlearned response to hold its breath when it goes underwater. These reflexes start to die off after a while because you get conditioned to different things. But at certain ages, they have these reflexes. So.
Yeah.
This is what that's harkening back to, is the fact that we can't help but go like and salivate and stuff like that when there's something tasty in there, and that's just the way that it works. You don't get to decide if your mouth salivates, for the most part when you're doing things. That happens whether you like it or not. So
Yeah, I know. Like, even though as I was reading, I was questioning it. Like this seems, but I also know, like, I read quite a bit of like psychological research,
yeah.
where it's like so I read also a lot of research like on attachment, and like that.
Mm.
The early research on that is just like observing child behavior and like how it responds to an attachment figure. And so it's like, and that, um, there's a lot of robust empirical research about that. So it's like,
Yeah.
I can't, even though as I was reading it, I was like squinting, like this seems a little bit sketchy. I also
Yeah.
know that like the observing behaviors, you can totally form conclusions about just observing behavior. Like we do it. Well, that is attachment theory research. I was just like, hey, how do you respond to attachment figure, etc., etc. And I read a
It's
lot.
also, that's the fun part of animal research. As much as there's lots of the stuff that's a little bit difficult to wade through, giving them a handful of banana flavored pellets or a handful of M&Ms and M's and watching them be like, "It's raining M&Ms. This is amazing." Is that's the glorious part of what we do, is we get to test good things too. We're not always torturing them,
No,
right? So
absolutely. And here, well, again, I'm sure like there's probably an abundance of research, just showing that yeah, M&Ms is like the most appropriate sort of way to get them to
they like
trigger.
them.
Uh, and I guess it's specifically just your stand. I wonder, has the experiment been run of M&Ms versus Smarties? Probably.
That's a very Canadian reference. The rest of the world will think that
Yeah.
Smarties are those little chalky things. But yeah, so what would happen is this would be pilot testing that they would talk to reviewers about, but wouldn't make the publication because you have word limits. But someone, like when I was doing place preference testing for mice and stuff like that to see, which area they wanted to go to, and if I could make them go to one area with a reward, I wanted to test which reward they find the most rewarding. So what did I do? I put apples applesauce, condensed milk, etc., in little piles and just looked which one did they go to the most. And I didn't put that in a paper. I'm never going to put that in a paper, but I had to do my due diligence because what if I'm not using the right thing? And that's the pilot work that just kind of never sees the light of day, but it's kind of implicit in this research. And certainly someone would have been like, okay, yeah, should we see if they want M&Ms or if they want the sucrose pellets or if they want whatever. Yeah.
Yeah, that makes total sense. And again, another thing, not to like nitpick or anything, but
Mhm.
I guess I can't help it. But like even here, so it's like comparing M&Ms versus Chow. Like it's not a binary, even though like even in reality, if you put Chow versus M&Ms, for me that's going to be a binary. And so it seems like there's like that small preference, occasionally once in a while, like for Chow. And I'm just like super curious as like what's going on there. Of like, hey, why do you get any Chow signal like what? So and I don't even
Yeah,
know.
I mean, exploratory behavior, satiety, they don't show the order that they're doing these things
True,
in. It
yeah.
might, they're measuring probably the chow. I would have to read their way that they measure it, but I presume that it's by weight. And that you measure it when you put it in there in the morning, and then you subtract how much is left when you go back 24 hours later. Because that's just the simplest and less labor intensive way to do it. You could have a student sitting there clicking and seeing how often they go to the thing, but that would be overkill, right?
Yeah.
You just they don't need to do that. And maybe they put the M&Ms in the cage. Maybe they put them in a little thing with M&Ms. I'm sure they describe it and I just don't remember it. But it could be an order effect. It could be the same satiety effect that we were talking about earlier with the salt. And some sort of mechanism that's just not controlled for otherwise, right? Because you can food deprive animals, or you call it food deprivation. That makes it sound a lot worse than it is. It's specific time feeding. So anytime that you're not giving them ad libitum access to food, which they do not have in nature, by the way,
Yeah.
anytime that you don't give them ad libitum access, it's called food restricted, or food deprived. And basically that just means that you can, in situations where that's important, you can control when they have their food and make sure that they are hungry when you're doing a test like this. And maybe they did do that. Again, I haven't read it recently enough at that level of detail to remember, but I've done experiments where my animals get fed when they're done for the day, and then they get ad libitum food throughout the night, and then I take it away about 4 hours before so they don't have access to it, so that I can control a little bit better for those things. So
Yeah.
basically I'm saying, like, yeah, you gotta wait till supper time, buddy, sorry.
And since again, I don't really know what this research looks like at the boots on the ground level, but I'm assuming this is like controlled for, like I'm assuming like the chow and the M&Ms are in a dish, and as you say, like they pretty much just like put the food in, and then they weigh it afterwards, and then they pretty much they calculate, well, this food is missing, that's what they ate. I have to assume, I'm sure it's a trivial thing of like it's not, some of it isn't lost, or just like they knock it off of like the tray, or like some pellets are like hidden in the cage, or like falls on the ground, something to that
Yeah,
effect.
so food pellets for rats are pretty big, bigger than the thing that I showed for their brains. They're like a cylindrical kind of thing about this long. They're pretty
Okay.
obvious. Even a half-eaten pellet amongst usually the bedding that they're in is cedar chips or ground up corn cobs to try and be more ecologically friendly, or environmentally friendly. Sorry. And then they get a whole bunch of fluffy bedding and stuff like that. But the point is, it's very different. They get like a little enrichment tubey thing that they hang out in. It's kind of like a hamster cage, basically, that they're in. If you picture like a pet hamster, and they get like a little thing, and there's a slanted part of the lid that they can like climb around on and stuff. Anyways, it sticks out, and they don't go through the slats of the cage
Okay, yeah.
unless they're small enough to do that. Otherwise, they would be falling through all the time, and it would be a nightmare in there, and they would be peeing on it and stuff like that. So yeah, it's just easy to distinguish in that way.
Okay, yeah, that makes total sense. And if you're able to, I know you didn't conduct this research, and I have no idea how often M&Ms are used in rotor research. But do you know if like, basically, are they whole M&Ms with like the shell, like they ground up, basically? Like I don't.
I believe they'd be whole M&Ms. They probably went to the store and bought M&Ms.
Okay,
Yeah, we probably wouldn't buy them from like VWR or something like that. But You do buy the sucrose pellets specifically from a company though, because they need to fit into the pellet dispenser. That's a very specific size and parameters, and
Right,
you
that makes
actually have to
sense.
sift out the dust so it doesn't get stuck. And it's a pain in the ass, to be frank. But yeah, it's probably just whole M&Ms from the store.
Yeah, I wouldn't have. Again, I don't. I know virtually nothing about rodents, but like, yeah, I don't know like their jaw power. But I guess it's pretty like. I just have a hard time imagining like a rat like biting into like a whole hard shell M&M. But obviously they can do it.
Oh, they can. They can. I don't know. They have a very powerful bite strength. They actually have to bite on very hard things like plastic and stuff like that because their teeth, their front two teeth, grow indefinitely like a beaver.
Oh, okay.
So we have to keep them ground down, otherwise they'll grow out. So if you only gave them soft food and access to soft things, they would suffer because their front tooth would grow into their chin.
Oh yeah, so they need to like voluntarily like grind their teeth down to like manage them. Okay, yeah, that makes
Exactly.
total sense. Well, the more you know. But it's like kind of
Yeah, these are things that I learned along the way that I'm like, huh, guess so.
uh,
Cool.
so yeah, I think the main message like here is like yeah, they're not getting like extra enjoyment from the food or like the sucrose, like through some other mechanism. I think that's pretty much like the overall conclusion, not one of the overall conclusions like of the paper. It's not an increase in basically pleasure or hedonic liking. It's just like something, well, sort of like as you say, they need something to focus their attention on, and then stimulation of that particular region in the brain, the CEA,
Hmm.
main message
Yeah.
of the paper. And I guess to wrap it up, I do sort of have some, I guess, sort of like macro, maybe like one or two
Hmm.
macro level questions,
Absolutely.
mostly driven by my own curiosity of like, basically, like in this research, like the way they carry it out, like they pretty like, I would say, they like conclusively show that like in the experimental like modified rodent, like with the light and the sucrose, like they're able to basically, at the very least, to some extent, cultivate drug seeking behavior, and it seems like sort of like unanimous like across the board. But then in terms of like translating it like to humans,
Hmm.
where it's like so you're way more well versed in addiction than I am. But like is it presumably something as simple of like this region of the brain, even though like generally even using the example, so I know your area of expertise or one of your areas of expertise is like opioids,
Hmm.
where it's like yeah, obviously there's like a lot of news about people unfortunately becoming addicted. But then like uh, what's curious to me, it's like from what I know of the data, and correct me if wrong, like the majority of people like don't become addicted to opioids, or at least when appropriate, like prescribed. It's not like a one-to-one, right, where if like you're exposed to opioids, you become addicted. uh
Yeah.
and so it's like here it's like it seems like almost like the rats have like no choice but to become basically seeking the sucrose as they've been conditioned, as opposed to it seems like more gray, or it's like it's not as automatic, at least when it comes to humans, at least of what I know of opioids. My
Yeah.
understanding well, incorrect or well, if you have anything to say, by all means go for
I
it.
have I have a great vast amount of things to say because that was the precise, broad question of my undergraduate and master's thesis research. So, as you say, most people don't get addicted when they try things. The vast majority of individuals have tried some addictive substance or another, whether they got painkillers prescribed when they got their wisdom teeth taken out, the caffeine that you drink every day if you're, usually an academic. If you don't, that you're an anomaly. Alcohol, lots of us have tried alcohol and we're not alcoholics, or basically the point is finding someone that hasn't tried an addictive substance is rare, right? But most of the people don't become addicted, right? The proportion is, I think, about 9% have some sort of diagnostic-ish criteria in a lot of the studies. And then globally, I think it's 2% pass the official diagnostic criteria by the ICE. Or IC, I can't
ICD, I
remember.
think, International
ICD,
Classification. Yeah.
sorry. Yeah. Yeah, exactly. The ICD. So that's not a whole lot. It's a huge amount of people because there's a lot of people on Earth, and those are presumably very underreported and stringent criteria. But the idea is that there's something. What makes these individuals different? And one of the most salient things, at least in the research that I did, was of course genetics. If you're genetically predisposed, that's one thing, and there's lots of genes that contribute to addictive vulnerability. But the other thing is your upbringing, your life experience, especially in vulnerable times and times where you're particularly malleable in terms of your neuroplasticity and that sort of thing, though your brain stays plastic throughout life.
Hmm.
um, and that's exactly the topic of my research in my master's. So we were looking at the effects of early life stress because we know that the proportion of adverse childhood experiences, as that goes from 0 to 1, 2, 3, 4, or more than 5, those numbers basically correlate almost perfectly with the incidence of addictive behaviors and problems with substance abuse later on in life. So there's definitely something associated there, but it's not it's not a nail in a coffin, right? Because there's lots of people that experience that and don't go on to become addicted. So it seems to be that there's this kind of perfect storm that needs to happen where you maybe have a slight genetic predisposition. And then if you have that genetic predisposition, and then that's the first hit in the 2-hit hypothesis model, to use the nomenclature, the second hit would be if you're unfortunate enough to have, a shitty childhood. And content warning here, like this can be anything from neglectful behavior on behalf of the parents to not going to school with a lunch,
Hmm.
to things as explicit as outright abuse
Yeah,
of any
absolutely.
kind, emotionally, psychologically, etc. um, and we know that these things seem to affect it correlationally in human studies, but doing that in rodents is something that we can do, and it's something that it's